Micheletti. I'm a professor in biochemical engineering, University College London, and co-director on one of the two UKVN-funded hubs called VaxHub. I'd just like to start to say a few words on how the idea behind the event came about, and acknowledge my friend and colleague Gilbert, who later last year proposed a lessons learned event at the back of one of the vaccine CPD courses we run in UCL. Me and Professor Sarah Gilbert, the co-director of the VaxHub, really liked the idea. And this gave us also the opportunity to work together with the Imperial Hub team and really further develop the original idea into today's event. We certainly didn't expect so many registrations, participants from very different sectors, from so many countries. Apologies to our colleagues in North and South America. We know the choice of timing is not ideal, but we hope we can enjoy the event once the recording is made available. The popularity of this event certainly shows that there is interest in exploring some of the questions we have set for reflecting on the role of academia, on the importance of collaborations, on discussions on global approaches, and I really hope you enjoy what we have put together. So I'm going to hand over to Ben for the introduction to the event.
Thank you, Martina. So I'd like to offer you a welcome from my side. My name is Ben Pierce. I am Operations Manager of the Future Vaccine Manufacturing Research Hub from Imperial College London. It's been my pleasure to co-organize this event with VaxHub, Martina and her team. It's been a delight to have so many prestigious speakers and panelists. And we'd like to give our gratitude to you, the attendees of today's event. And where we'll be focusing on vaccine manufacturing during a global pandemic, what we have learned from COVID-19. But I do want to begin with some housekeeping notes. And just before we get to the session one, race against the clock. Please be advised that the event is being recorded. You have been notified of this beforehand, but the recording began just a few minutes ago. We encourage everyone to enter any questions in the Q&A section at the bottom of your Zoom menu. For the Q&A sessions at the end of each section, we have early career researchers from our hubs as co-chairs who will be selecting and collating questions that will be presented to the speakers and panelists from that session. We will also be presenting some poll questions throughout. Please do let us know your feedback. We're very interested to hearing what your responses to our few poll questions will be. And we would also like to offer our great thanks to our funders, the Department of Health and Social Care. And we have these hubs set up through UK aid funding that's being managed by the EPSRC, a research council within the UK, now part of the UK RI. We would like to point out that the views and opinions that you may hear today are not necessarily those of the Department of Health and Social Care and our funders. But we do encourage you to highlight the event on social media. We've created a hashtag. Please use the hashtag shown at the bottom of this screen to circulate interesting points and outcomes, questions, thoughts, opinions about the event. Very, very much looking forward to seeing it being circulated from today. Now, turning back to the Department of Health and Social Care, I would like to point out how the hubs have been funded and how they began. From 2016 to 2021, the UK government made the decision to commit £120 million of UKA funds to support research for vaccine manufacturing, vaccine development against pathogens of particular importance in low and middle income countries or developing countries. And a stream of research activities was generated through that under the guidance of the UK Vaccine Network, or the UKVN. You may see that acronym throughout today. And from that, two innovative future manufacturing research hubs were born in 2017, but really began from 2018. And just to avoid any confusion, One is called VaxHub, and that's from the University College London, co-directed by Professor Martina Micheletti and Professor Sarah Gilbert. And the hub where I'm a part of as operations manager is called the Future Vaccine Manufacturing Research Hub, but for brevity for today, I will be calling it the Imperial Hub throughout. That's of course from Imperial College London and directed by Professor Robin Shattuck. So these are the two hubs that will form the consolidated summaries of activities that you'll be presented with today. And just note that it is just under 17 million pounds and a relatively small portion of a vast amount of research that has been allocated from this 120 million pounds through the UKVN stream. This is not an exhaustive list of all the projects. There are some others, but just note that with the announcement from November 2020 that the UK is cutting UK aid from 0.7 to 0.5%. That's a small number in terms of percentage, but it amounts to nearly 4 billion pounds. We may see severe impacts and more barriers to our engagements and support for vaccine research, not only globally and particularly in developing countries, but also with severe impacts likely in the UK as well. So with that brief introduction, I'd like to hand it back to my colleague, Martina. Off to you, Martina.
Can we have the next slide, please? Thank you. It is my pleasure to present the agenda today. We will start with reflecting on the race to produce COVID-19 vaccines and really on the research behind these efforts with some of the UK key players. Ben will then be your chair for the next two sessions, which will focus on the hub's role in supporting global manufacturing capacity building. We will then move to discuss pandemic preparedness, and then in the end, the hub directors will draw some conclusions on the lessons learned and highlight what should be the focus in the future. Just a reminder, questions are going to be handled after the last speaker for each session. So please do use the Q&A chat. And if you wish, please name the speaker the question should be addressed to. We have also set a few polls to engage with the audience. They're going to show you on your screen, so please answer if you wish using the poll function. I think the first one is our icebreaker and is shown or should be shown to your screen shortly. I'm now going to present our first speaker. It's my pleasure to introduce our keynote speaker, Professor. So Steve Bagshaw, he has over 35 experience across the biotechnologies and fine chemicals business and manufacturing sectors. He was CEO for 6 years of Fujifilm Biosynth Biotechnologies. Steve chaired an important board, the UK Industrial Biotechnology Leadership Forum, also co-chair of the UK Bioeconomy Strategy Board. Is currently working with the UK Vaccine Task Force as an industry advisor, is non-executive director of CPI, and has been a council member of one of our councils, the Biotechnology and Biological Sciences Research Council. His efforts were recognized by many awards. I'm going to mention one which is particularly dear to me, which is the Peter Daniel Award for contribution to UK bioprocessing. Over to you, Steve.
Great introduction, thanks, and dear to me too, that award. That's lovely that you should say it like that. I got a chance to look back at the way in which the VaxHubs were set up back in 2017, and as I read the stories that were written back then and the applications, what struck me was that it was talking to me from a land that was like a foreign land almost in terms of where 2017 was compared to where we are now. In 2017, of course, pandemic was something that we were talking about as lessons learned from SARS, from swine flu, from MERS and Ebola. Whereas today, we're all significantly wiser in what it means to deal with pandemic, with global scale manufacturing, and the need for effective vaccines. What I'm hoping is that in this event, you'll get a chance to look back, but also you'll use the lessons learned in a way in which we've done so far from 2017 to today, but accelerating them so that the next 15 months of pandemic is informed in a way in which the first 15 months only started to be informed. You can see that as you start to get into this, and you see society changing on our watch. Martina talked about the years in which I've been working in biopharmaceuticals. And throughout that period, whenever I tried to explain to somebody what exactly I was doing, you could see their face glaze over as you got to biopharmaceuticals. You didn't even have to start talking about manufacturing. It was just enough, the name. If you added advanced therapies or even got to vaccines, no one really was still listening. Now what I've found is a complete difference. People understand that if you're working on vaccines, it's important. And last year, as those of you who are following me will know better than I do, everybody was interested in what happens next. The question on everyone's lips was, will we have a successful vaccine? Will we be able to create something that's safe? Will it come by Christmas? Would you take it? These were the questions that many of us were getting asked. No matter how small or big the role was you were playing in vaccines, if you said you were working on the vaccine, then you were subjected to questions and answers. What I find now as I walk around is that when people ask me the question, what do you do? And I say, well, I work with the Vaccines Task Force, almost immediately their answer is, well, thank you. Because there is such a change in the way in which people now perceive the role of vaccines and in the way in which normal life, whatever that means, can come in the future, and how dependent we are as a globe on successful deployment of vaccines. I was embarrassed the first time someone said thank you to me, because I realized what a small role I was playing in the story. But what I realized as more and more people said it to me was that I needed to pass that on. And so I pass it on to you, the thanks of my neighbors. My neighbor who texted me two nights ago to tell me that her husband and she had just had her second dose, and she said thank you. And so I pass on her thanks too. And I know that there are people around the world who would join me in saying thank you to all of you who played some part in the development of vaccines to deal with COVID-19. For many of you who would describe yourselves as scientists, you've also seen another change, and that is that society sees scientists as trusted voices in this pandemic. Some of you who've been exposed to the press may not think that, but as a whole, the scientific community is seen in a better light now than it was, and is one of the trusted voices of what happens next. What should we be doing? What does the science say? Who would have believed that there would be headlines like the Deputy Chief Medical Officer having to turn down Strictly Come Dancing? It just wasn't something that anybody thought of 12, 15 months ago. And yet his has been one of the trusted voices throughout this pandemic, and someone that people listen to. And so for many of you who've been on the radio and who've been on television, I also say a huge thank you for putting yourselves through that and explaining to people just how it is from your aspect and how you see the science. Because not only are you explaining it to people, you're also inspiring a generation of people who are thinking now that science matters and science is cool. And that means that young adults, children, and particularly parents who are helping their children make decisions are seeing the option of taking on science as being something really important. And in the long term, that's a fantastic thing for us all. The issue with COVID-19, whenever we come and talk about it, is that the stories that we tell often start to get personal too. So all of us will know someone who knows someone who's been affected by COVID-19, a life lost, someone who this time last year was with us and who isn't with us today. And that's still going on all around the world. And you see that in your pictures on the news. And anybody who says that this crisis is nearly over is not really looking beyond the end of their nose. There is so much still to do in this area. And so all of what we're going to talk about today matters, and matters immensely in a way that back in 2017, many of you who signed those papers knew it would do one day, and it does now really matter to the world. So let's talk about the response of how we responded in this country to the COVID-19 crisis. And I'm being followed by two eminent speakers on this, so I'm not going to spend too much talking about the way in which Oxford and Imperial worked. I'm going to talk more in general about how we as a community responded. The government appointed a task force at the highest level, responding to the Prime Minister back in April last year, and that task force brought together experts from industry, from academia and from government, and looked at ways in which they could somehow plot an end-to-end journey to get to mass vaccination in this country. Given three targets. One was to get access to the most promising vaccines for the UK population. Second, to make sure that they were available internationally. And thirdly, to put legacy in place such that we were better prepared for the next pandemic. In bringing together the academics who were working on that, we realized that in hindsight now, that there were decades of experience being sought and used and worked with in that collaboration, both as individuals and institutions. And that through the period of the past 5, 10, 15, 20 years, that had also been backed by, although you may not feel so when you're in the funding rounds, a consistent level of investment, such that institutions and individuals were able to have careers and institutions were able to grow in terms of the way in which we address vaccines and vaccine development. Dame Otterline described it as being a flexible, agile, empowered, interconnected network of innovators, scientists and researchers. And that when you bring this together in the UK, you have something special, something that is very much a national treasure and something that needs to be invested in. And I'm sure as we get to the end of this and look to the future of where initiatives like this go, that will be another piece that we come back to. And yet in a pandemic crisis, that isn't enough. It isn't enough to have something that has as deep an expertise as that. It has to be joined up, because end-to-end acceleration requires us to break down barriers in all sorts of directions. And so what the Vaccine Task Force did was to bring together expertise, not just from academia, but also from industry and also from government. And it sponsored conversations and the chemistry between individuals. You'll have read in the press of CEOs who met researchers and recognized there and then that they needed to act, and were able to bring the might of an industrial powerhouse behind certain academic institutions or ways of working. And though we may look back and think of some of that as serendipitous, we will also see that around the world, governments, academia and industry realized that they were better together than they were apart, and that process acceleration would only come when those relationships started to also be turned into some kind of process. And so the Vaccine Task Force spent significant time through the summer of last year doing due diligence on hundreds of promising vaccines, getting to a list of multimodal vaccines and a recommendation of which were the most promising and which were the ones that should have deals done with them. And you know this across adenovirus, across the mRNA, across the inactivated virus, across protein subunits, across plant-based, across some of the antibodies, some of the treatments that are out there, all of those being assessed and all of those being backed then by either a deal or investment. Rapid scale-up, process establishment, qualification and approval was all happening in parallel with that due diligence in the different supply chains associated with each vaccine. And what this government did, and the Vaccine Task Force recommended, was that capacity was reserved, capacity was invested in. So you had Oxford Biomedica, Wockhardt, Cobra, Valneva, all invested in. Then you have Fujifilm Diosynth, GSK and CPI also with capacity reserved and investments made. Facilities that had been commissioned, like VMIC, were re-energized or not even re-energized, just energized and made to come quicker, but with a much bigger scope. As were some of the investments made at Porton Down and some of the testing facilities. and across the network, and a repurposing of assets such as the one at CGMIC have now. That's the Cell and Gene Therapy Catapult at Braintree, where large-scale commercialization can be done of vaccines that no capacity was available to be done in the UK, and now can be done in Braintree. Alongside that, skilled people were secured, and supply chains that, known to be global, backwards and forwards up the line, were looked at, and partnerships, relationships made to try and secure all that was needed to make sure that you could scale up successfully anything that was a successful vaccine. In the early days, we like to think of not dropping the ball. We were putting around people a set of supply chain experts who would just listen and say, so you've got a problem with bags, let us sort that out. You've got a problem with that solution or that test, let us try and sort that out. That was the kind of support that was happening in the early days. And I'm delighted to be sharing the platform today with two people who were carrying the ball at that point, and we were in the vaccine task force trying to support them carrying that ball. But as well as making sure you make population scale doses, you have to have them approved. And so the clinical trials processes were also being revamped and looked at. And so a registry was put together with 500,000 people volunteering to be involved in clinical trials going forward, to try and shortcut the time it would take to set clinical trials up. Similarly, in terms of the regulator deploying rolling submission of data, parallel analysis, and getting to the end, so that if you think of that project plan that traditionally goes like this, with another item and then another item and then another item, and the date just keeps going right across the page. In this, the date was drawn left across the page, and the regulator was able to, within days of being given the last piece of data, have an opinion on how approvable the product was. These are things that we'll look back on as ways in which we transformed the way in which you could get clinical trials done and get to approvals. But even now, if you approve a vaccine, you've still got to deploy it. And so in parallel with all of this, starting in the summer of last year, was the NHS efforts to make sure that we had the largest mass vaccination rollout that we've seen in this country. And so the NHS, working with their own professionals, but then across the nation, and all of you will have enacted in the UK, will have encountered somebody somewhere who's volunteered, who's played a part in either vaccinating or being one of the welcomers or running the phones or doing some part of that last mile of getting the vaccine from the warehouse into the arm of the people. And so all of that's happened in parallel with a huge civil service activity to remove roadblocks and to get funding. And so that trio of government, academia and industry has been a real success story of how you bring civil servants alongside academics and industry, working within the funding mechanisms that there are and creating new funding mechanisms where they were needed to actually build the infrastructure that was needed to roll out the vaccine. So where we're sat today is that we've got 2 billion doses out globally. We've got 69 million doses in the UK that have been given out. It's not over. It's not enough. We need to keep going down that track. However, pause for a second and say, a year ago, would you have predicted 2 billion doses by June the 10th next year? That's as long as you get in the vaccine world, though. That's the pause. You go, yes, great work, but look at how much more there is to do. So let's just quickly finish then by talking about the things that we've got going forward. Continuing manufacturing and deployment, promising vaccines in development. There are still vaccines that we need to get to market. We need to get them because they're the ones that will help us get global scale production. There are partnerships to deal with variant that we're undertaking. We're looking at more investment in sequencing, in testing, in clinical trials to help us, and embedding the learning from the last 15 months. And looking forward, we need a deeper and more vibrant, a more resilient vaccine ecosystem to be prepared and more ready for next time. And that ecosystem is being developed by working with developers, bringing in larger scale looking at the larger scale manufacturers and looking at what would it take to have vaccine manufacture here in the UK. We're looking at supply chains and how we can bring those onshore. We're looking at technologies in terms of innovation and research around manufacturing, formulation, and clinical studies, and looking at the ways in which we can improve deployment, and very much taking some of the echoes of the VaxHub work, and the cross-government, making sure this is joined up. Too often, many of you will have been subjected to the fact that there are people who work with you on one line, but no one knows what the left hand's doing or the right hand's doing. And so there's a real piece of work being done to try and join up our response. To me, this is the tribute to the past 15 months that we're paying here, and to the past 3.5 years of the VaxHubs. It's a positive legacy that comes out of the work that was done by yourselves and what's happened in the last 15 months. It will help us with the next 15 months, but more importantly, it's also got to keep us ready for the next one. Because one thing 2017 to 2021 has taught us is that we never know when the next crisis is going to come, but it's likely to be sooner than we think. And with that, I say thank you.
Thank you, Steve. Thank you for highlighting all the work of the Vaccine Task Force, but especially the role of science in the society and the impact on the next generation. I think we're going to explore some of the points you made in the Q&A session later. So for all attendees, if you have questions for Steve, please post them in the QA chat and we'll discuss with Steve later on this morning. So I'm going to now introduce our next speaker. It's a pleasure to welcome Professor Robin Shattuck. He's the head of mucosal infection and immunity within the Department of Medicine at Imperial College London. His research focuses on the mechanisms of mucosal infection and the development of novel preventative strategies appropriate to a developing world setting. So he has secured funding from very different sources. I was going through all his projects, many interesting ones. Gates, EU, Wellcome, NIH, MRC are some of the funders, and is the director of the Future Vaccine Manufacturing Research Hub at UPL. Over to you, Robin.
Thank you very much, and good morning, everybody. So welcome this morning. I'm going to be talking to you about one aspect of the Future Vaccine Manufacturing Research Hub that we have been focusing on, and really started working on this back in 2017 with the view that pandemic threats was something that should be taken seriously, and we needed to have new technology to prepare for such an eventuality. I'm not quite sure. Oh, there we go. And so we were already looking at RNA technology for a number of reasons, partly because it allows you to manufacture on a very low footprint with small capex. It allows you to do rapid production and to make potentially millions of doses within weeks, and meets that just-in-time response capability for Pandemic X. And we've kind of put this forward as an approach, a new approach for pandemics at the World Economic Forum in 2018. And I think we had about 30 people turn up to listen to the talk, and they probably got lost and just turned up off spec rather than deliberately searching us out. Because at that stage, people were saying, well, you've talked about pandemic threats, and people have been mentioning this for the last 10 years, but it never seems to quite happen. Of course, hindsight is a beautiful thing. But one of the reasons that we were very interested in this technology is really this small footprint approach. And we had the vision, and still have the vision, that you could set up manufacturing sites in the equivalent space of 2 shipping containers. And it would be paradigm breaking in that rather than having to have large warehouse-sized manufacturing facilities that supply the world, you could have network facilities around the world ready to build RNA And then the design of the vaccine becomes more like the software. So in the same way that the personal computer allowed access to information and the internet, we think that in the future, small-scale manufacturing may allow global access to be more readily built for future pandemics. And certainly one of the things that we're seeing as a result of the COVID-19 pandemic is an explosion in countries interested in wanting to set up mRNA manufacture in different parts of the world. So this is definitely the way the world is going. And we were really starting to develop this from the beginning. Well, just to give you a quick tour of the two types of RNA vaccines that are out there at the moment. There is conventional mRNA vaccines, and you'll have heard of these because these were very successful, both from Moderna and Pfizer, getting emergency authorization, and millions of doses of these vaccines have now been administered. These are relatively simple. They encode a gene of interest, and in this context, it was the glycoprotein of SARS-CoV-2. They need to be encapsulated in a lipid nanoparticle to gain entry into the cells, and when they're in the cells, they carry the code for the spike glycoprotein and engage the cellular machinery to make that protein. So if you like, it's moving away from manufacturing a complicated vaccine, either recombinant proteins or virus, and really using the cell's machinery to make the vaccine itself. Well, we have been interested in self-amplifying RNA, and the reason for this is because it allows you to use lower doses. Self-amplifying RNA is based on, typically on an alpha virus backbone, where we use the non-structural proteins that encode the polymerase that allows the RNA to be amplified once it gets into the cell. Now, it has commonalities. It also needs to be encapsulated to deliver it into the cell. But once in the cell, its polymerase machinery makes multiple copies of the RNA. And so essentially you get more protein out with a lower input of RNA. We think this is going to be important in the future because you can look in terms of the doses of RNA that's required for Moderna, 100 micrograms per dose. Pfizer-BioNTech is at about 30. And we are targeting a dose of 1 to 5. We still our ideal is to get down to 1. This is important on a number of issues. Firstly, it allows you obviously to manufacture much more doses for the same volume of manufacturing material. It also means that the safety profile is better because there is some side effects with higher doses of RNA. But perhaps more critically, it also allows us to think about combination vaccines, where if you were looking at a combination of, say, influenza, 3 influenza targets and a COVID-19 target, you'd be looking at a 4 microgram dose as opposed to a 400 microgram dose. So we also see this as very much technology of the future for multi-component vaccines in the RNA space. Well, we started our journey in January of 2020 by engineering spike glycoprotein. And like many, we used data from the literature. We were able to make a prefusion stabilized version of this. We thought this was important to maximize the induction of neutralizing antibodies. And we were able to move with support from the VaxHub, the Vaccine Hub, in terms of funding into preclinical models. We saw that this vaccine was very, very potent in small animal studies. And then more recently, and perhaps Sarah may touch on this in her talk as well, we were able to see, again, in small animal studies, that it's very complementary to other approaches, and it can either prime or boost viral vectors. And this may be important in terms of thinking about how these technologies are used in the future, where one can mix and match different vaccine candidates. And there are some obvious advantages there, both in terms of enhancing immune response, but also availability of vaccine doses. Well, we were working on the manufacturing process in our hub at the beginning of the epidemic. And really at this time, it was very much laboratory scale, looking at the work to start to do process innovation and scale up when suddenly SARS-CoV-2 appeared on the horizon. And so we had to work out very quickly how to manufacture a vaccine, having never done this ourselves from an academic background, rather than coming from it from a well-formed commercial entity. And what we had to do was work out how to go from sequence into production process to rapidly produce clinical quality grade RNA and to formulate it. Now, to do that, we had to make some very quick judgments and decisions. And so we had to take off the shelf a formulation that had been developed for messenger RNA. In fact, it's almost an identical formulation to the one used by Pfizer-BioNTech. And so we were taking a technology that had never been into humans before. So a very different approach to any of the other vaccines that you've heard of that are out there. We were going from sequence with a new technology to first in human studies in a record-breaking time. Now, of course, we didn't have the advantage that other RNA companies had, that they had had 10 years worth of development time and several clinical trials before they embarked on this journey. And so in some ways, perhaps we were really pushing the envelope very fast in order to get an early clinical signal. Now, in the context of the pandemic, you have to remember that when we started this journey in January, this was not a pandemic. It was still an isolated outbreak of a new virus in China. And we were thinking at this stage, well, we might be developing a vaccine that, if it was successful, might be used in China, rather anticipating that we would be looking at it in the context of a global pandemic. And of course, the attractiveness of the approach is that we could potentially produce, within a very small reaction volume, millions of doses. And that's still something that is realizable. Well, what we were able to do is to move from that sequence. In fact, we didn't start designing a vaccine until the 19th of January. We'd sat on the fence. Seems strange now to think about that. We were able to assemble in very quick time a vaccine and get it into preclinical animals. Again, this was really only achievable because we had funding through the hub system, and that allowed us to get into a preclinical toxicology study without having to wait for grant funding from peer review mechanisms. And in fact, one of the lessons we learned was that the peer review process really was very slow, and if we'd relied on that exclusively, we wouldn't have made the timelines that we were able to realize. Now, because we weren't a company, we had to very rapidly look for CMOs to manufacture our vaccine. In fact, we had to turn to a company in San Diego, TriLink, to manufacture the RNA, and another company in Austria, Polymune, to formulate the material, working with a Canadian company on the formulation, to supply the formulation for that process in Polymune. So again, in terms of looking to the future, we definitely need to build that capacity within the UK to allow this technology to move much faster. But we were able to get into the clinic by the middle of June with a technology that had never been in human studies before. And I think that was probably not the fastest in terms of vaccine development globally, but the fastest that anybody has ever done that with a brand new technology. Well, the rest is history, as it were. We went very quickly into dose escalation studies. Remember, we had no idea about the potency of this vaccine in humans, so we had no reference, unlike the other technologies that had a reference of previous clinical trials. We went through dose expansion and a large safety trial involving over 400 participants. And we were planning to move into efficacy studies this year, but events really overtook us because of the successes of the other companies. Now, that's a fantastic outcome. You have to remember, again, early in last year, people really didn't know whether any of these vaccines would work. And so it was important to have as many strategies moving forward in case only a few came through with a successful outcome. But in fact, the really great news is that many vaccines came through with a very high bar of efficacy, particularly the work from Moderna and BioNTech. I won't talk about the adenoviral vectors because Sarah will cover that very nicely in the following talk. So our vaccine was shown to be safe and immunogenic. I can't show you the data. We've submitted those for publication, and we hope that that will appear on a preprint server literally in days. However, we took the view at the time that when we got the data that it wasn't as good as those vaccines that were already out there with our first generation approach. And so we have been re-engineering, redeveloping and improving on the vaccine, and we have other trials, which I'll talk about in a moment, that are moving forward. Of course, other people's success have made it more problematic for us to move our vaccine further forward in terms of it makes those efficacy trials more difficult. And so, for example, we've started a new vaccine study, and it's becoming quite complicated to find people in the UK who've not already received vaccination, who want to receive an untried novel vaccine approach. We also recognize very much that, in fact, we lacked pharma backing, and that was something that really set us back, perhaps, in terms of moving forward. And so we've been working to change that. And perhaps in the questions, we can come back to why pharma was less willing to take a bet on this new technology. However, we see that that's changing. So in terms of what we were able to do, we were able to advance this approach by probably 5 years. We now have variant vaccines that we have developed. We're looking at potential boosting strategies with different funders. And we are focusing technology very much on other indications, because although this has been very much about COVID-19, the technology is very much a platform technology. We were very much welcome the support from MRC UK, from NIHR and the Vaccine Task Force to get into clinical trials. But again, what we noticed is, in fact, we would have not been able to do the job without philanthropic donations. And this was really probably matched to the government support that we got. And this allowed us to progress without that stop-start scenario that you get with conventional grant funding approaches. So for the future, in terms of pandemic response, perhaps different funding mechanisms need to be considered. And one of the things we recognized very quickly looking back is that, in fact, we were really able to keep up into this early part of the clinical trial program. But once it moved beyond that, we were really falling behind based on investment. And you can see that the large levels of investment that the RNA companies got in order to get through efficacy and be first to market, compared to the investment that was provided for the Imperial program. That's not really to complain, but what I think it highlights is that the companies in Germany and the US that were able to respond in the RNA space actually had very strategic investment from their governments, both during COVID-19, but also in the preceding 10 years. And I think if we want in the UK to be an important player in the RNA space, there are some significant lessons we can learn from these other countries in terms of their investment in innovation and manufacturing in the RNA space. Well, one of the things we recognize very quickly is that RNA is really at the start of its journey. And one of the strengths, but also defects, is that when RNA enters into the cell, it's recognized as, by the innate immune system. This provides an adjuvant property, but it also serves to reduce the expression of RNA, because part of that antiviral approach is to reduce expression of RNA within cells that have been alerted to RNA being introduced into the cytoplasm. So another approach that we were able to develop with funding through the hub and also funding for Anna Blackney through a Marie Curie Fellowship, is that we were able to start to look to the future. And this represents work where we looked at inserting small components of other viruses that actually dampen that innate recognition and switch off the antiviral pathway, and encoding that into our vector. What we identified in this initial screen were two small proteins, ORF4A, interestingly from the MERS coronavirus, that actually shields double-stranded RNA from being recognized by some of the innate pathways that trigger an interferon response. And using funding from Innovate UK, we've now taken this into a new clinical trial that's just started this month. And we're hoping with funding through our hub to start a trial in Uganda. That's dependent on us getting a no-cost extension. But if we're able to realize that, that will be the first study of a self-amplifying RNA vaccine in the African continent. And so we're keen to be able to do that, both to demonstrate safety and look at response to such a vaccine in a different population. The other thing that we learned through this journey is that the way you present the RNA is particularly important. I mentioned that we had to take an off-the-shelf formulation. It hadn't been optimized for self-amplifying RNA, which is different to mRNA. And what we do have in the UK is a wealth of expertise, not just within our hub, but wider in academia, in formulation, in lipid nanoparticle approaches. But at the moment, it's very fragmented and not coordinated. And so a way of harnessing that approach for the future will accelerate UK's competency in this arena. Some of the things that we've been focusing in our hub are ways to move from ultra low cold storage of RNA vaccines through to storing at 2 to 8 for over 6 months and to room temperature for months, particularly work in our hub that's been led by Ranjan Chen and Jason Hallett, but also working with others outside of the hub. And so we think that very readily in the future, although RNA technology was limited for the pandemic by the cold chain, we will be able to solve that issue moving forward. The other thing that we were...
Hey, Robin. Sorry to interrupt. This is all very interesting, but we have a lot of questions and I want you and the others to have an opportunity to answer. So if you could...
I will wrap up.
Thanks a lot.
Very close to the end. So we also have been investing in manufacturing through CPI, partnering through the hub, and then picked up by the Vaccine Task Force, and quality control, a very important aspect of RNA vaccines that still needs to be resolved. And so we've been developing a pipeline of RNA, and I think the RNA approach is really very open to this. We have clinical trials funded by Innovate UK, funded by the CEPI program, and other partners moving forward into the future. And we think we're really very much at the beginning of this journey because RNA technology not only can be applied to infectious disease vaccines, oncology, but also a whole host of other indications. And so we've set up a spin-out company in order to support that. We're looking hopefully for other commercial partnerships. But I want to finally end on really we need to learn the lessons from other parts of the world that actually if we want to be players in RNA technology of tomorrow, we need to make ensuring that there is investment and forward thinking to make sure that it's available and invested in today. And I'll thank you there.
Thank you, Robin. And yeah, thank you for sharing the journey, but also highlight some of the challenges and some of the needs in terms of wider UK manufacturing capacity. It's a pleasure to introduce now Professor Sarah Gilbert. So she's a VaxHub co-director from University of Oxford, a professor of vaccinology, co-founder of the company Vaxitech. Her research focuses on development and testing of new vaccines designed to induce strong and protective T and B cell responses against many different viral pathogens. She has worked on influenza, malaria, Nipah, MERS, Lassa, among others. And as you probably all know, she led the efforts that brought us the COVID-19 vaccine in collaboration with AstraZeneca. Over to you, Sarah.
Thank you very much, Martina. pleasure to follow on from Robin. Presentations from each of the hubs showing what we've contributed to the pandemic, which I think you'll agree has been quite substantial, and it was very good foresight to have these hubs working in different technologies, but both providing very useful outputs. So I want to take you back to the Ebola outbreak in West Africa in 2014 going into 2015, because this was a catalyst for many people to realize that we really should be doing more to develop vaccines against pathogens that we already know about that can cause outbreaks. Now, Ebola had previously been identified, I think it was 1976. It had caused a number of small outbreaks. Somewhere in West or Central Africa, there's probably an outbreak every year, often in very rural, isolated areas, contained by contact tracing and quarantine, and there hadn't really been a great deal of effort to develop a vaccine against it. But what was different in 2014 was that it got into densely populated areas, into the cities where there was more opportunity for the virus to transmit between people. It spread between countries and rapidly developed into a much larger outbreak than ever been seen before. And at that point, we really needed a vaccine, and there wasn't one available. Now, some work had been done mainly in the US by a defense program to develop vaccines against Ebola to be used as a bioterrorist weapon potentially, but not really thinking about rollout in countries that might experience outbreaks. And so there was a lot of work to quickly bring on that vaccine development and to test vaccines in West Africa to see if we could then have a vaccine to deploy to end this outbreak. And although there were 3 countries mainly involved, Liberia, Sierra Leone, and Guinea, there was spreading to other countries, small number of cases in other countries, starting to see some international transfer as well. This was a much more slow burning outbreak than the respiratory pathogen SARS-CoV-2, but it did have the potential to spread much more widely. And so it was recognized that something needed to be done about this. I was involved in the early trials of one of the vaccines that had previously been tested in non-human primates and shown to be effective. It wasn't quite ready to go into clinical studies. We accelerated that. We got the vaccine put into vials, ready to initiate clinical studies. We did those in Oxford. We transferred data and vaccine to Mali, one of the neighboring countries, to the outbreak area. A further trial was done there. And we then expected that to go into an efficacy study to see if the vaccine actually worked. We knew that the reactogenicity was acceptable and that immune responses were being generated, but would it protect people against Ebola in the way it protected the macaques that had been immunized and then deliberately exposed to Ebola? What happened was that there was quite a long delay between getting that early safety and immunogenicity data and initiating an efficacy trial in West Africa. And while the plans for the efficacy trial were being discussed and the ethical way to do this trial was being debated, the incidence of cases was declining. And if you're going to test vaccine efficacy, you do have to have cases in which to determine that the vaccine's actually working against. And by the time the efficacy trials were ready to start, only one vaccine actually was tested and went through that process, and that was Merck's VSV vector vaccine expressing the Ebola glycoprotein. Fortunately, it had extremely high efficacy, and it was then able to be rolled out at the very end of the outbreak, although in reality, the outbreak had really been contained by contact tracing and quarantine, and the vaccine was just useful for mopping up a few small hotspots at the end. The difficulty of using this vaccine was that it requires ultra-low temperature storage, minus 80 degrees, and the other vaccine that we'd been involved in testing, the GSK's Chad3 vectored Ebola vaccine, didn't require ultra-low temperature storage. It's 4 degrees. It has a much more mature manufacturing process, could be made in very large numbers of doses, but it didn't get into an efficacy trial, and so therefore wasn't licensed for emergency use because there wasn't proof that it would actually protect against Ebola. So the Ebola outbreak and the control of it prompted a lot of looking back and lessons learned, as we're doing now, following the SARS-CoV-2 pandemic. And the WHO set out their R&D blueprint, encouraging vaccine developers to develop vaccines against the known pathogens. And the technology that we wanted to use for this was our ChAdOx1 replication deficient adenoviral vector vaccine, which is another platform technology, very adaptable, you have the technology that you do a lot of development work on, understand the manufacturing, the testing, the dose to use in humans, the reactogenicity, the immune response that's generated. And with all of that data on the platform, you can then very rapidly go into producing a vaccine against a number of different diseases. Now, for outbreak pathogens, where there's not any market to make money out of selling these vaccines, there may just be stockpiles, there may not be widespread use, but we have to be ready to use them if there is a large outbreak and try and prevent that outbreak from spreading more widely and potentially becoming a pandemic. It's very attractive to have a platform technology because a lot of the costs are associated with the development of the platform rather than the development of the individual vaccines. So once we've done the development on the platform, we have to spend less time and, importantly, less money on development of any of the individual vaccines. We do, of course, still need the clinical trials. We need to show safety, immunogenicity, and efficacy of all of those different vaccines, but the huge amount of work that's required on manufacturing, quality control, logistics of distribution of the vaccine, that applies to the platform, and it's already been done. So it's a replication deficient simian adenoviral vector vaccine. That means it doesn't spread through the body. It doesn't get attacked by any antibodies that people have already got through exposure to human adenoviruses, which is a common thing to happen. And we'd already got quite a lot of work done on the use of this vaccine in clinical trials, including, importantly, in a trial of a vaccine against another coronavirus, Middle East respiratory syndrome. So we took the approach in the Jenner Institute of using this technology widely and starting a whole list of programs. You can see the different diseases on the left to make the vaccines and work through what we needed to do to get them tested. So we've been doing this in 2019 and continually adding to this table. And the MERS vaccine development was highly relevant to the SARS-CoV-2 pandemic. Just a bit of background on MERS. Most cases occur in Saudi Arabia. It's endemic in camels in that population. It can spread from camels to people. They have outbreaks in hospitals from time to time. And there was one very large outbreak in South Korea, which involved 4 hospitals. It's only fatal in people who are older or immunocompromised, but there are infections in younger people, usually asymptomatic infections. So the fatality rate is probably overestimated because all of those mild or asymptomatic infections are not being detected. But the plan for this vaccine was to have a vaccine that could be used as a stockpile in case of a large outbreak, to vaccinate the healthcare workers in the hospital who themselves can become infected and then be an important means of transmitting the virus to other patients. A vaccine for travelers, a vaccine for people who work with camels, and also the idea of vaccinating the camels, because this virus has only been present in camels in the Middle East for about the last 50 years or so, and the incidence there has increased in the camel population. If that could again be reduced, then the problem of MERS in the Middle East would go away. And we had completed a phase 1 trial of the vaccine in the UK and had one underway in Saudi Arabia. So we then drew on our experience of the MERS vaccine and the development of the Challux-1 platform to make our COVID vaccine, again, the spike protein from the coronavirus put into the Challux-1. We did our first manufacture in Oxford, and these are the vials of vaccine that we made and had available by April to go into our first clinical trial. We took blood samples, we did lots of assays to measure the immune responses, and this is the overview of what happened in those trials, going from the vaccine design in early January 2020, the first human trial in April, and then moving into late stage trials in the UK by May, and then moving into trials in other countries. Oxford sponsored trials in Brazil, South Africa, and then in Kenya. This is because for a vaccine that we need to use around the world, it's extremely important to have data on how that vaccine behaves in different populations around the world. We can't just rely on data from one country. We have to look at it in different geographies, different ethnic populations, and that was the reason for starting up these other trials. We partnered with AstraZeneca. In April, they took on the manufacturing and very early on started large-scale manufacturing in many different sites so that there would be vaccine available for the eventual supply. They also sponsored trials themselves in the US, in Japan, and in Russia. Serum Institute did a trial in India, and by December of 2020, we had the authorization for supply in the UK. So I can skip that one. The efficacy data that came through in November were from the clinical trials run by Oxford, sponsored by Oxford. And of the 4 trials, initially we only had 2 of them included. That was a large-scale trial in the UK and the large-scale trial in Brazil, because at that point, the trials, the phase 1 trial in the UK and the phase 2 trial in South Africa hadn't had enough cases in the control group to be included in the analysis. We then added more data from those trials in subsequently. But initially what we were looking for is symptomatic PCR positive COVID-19 with one of those symptoms that are named here in people who had been seronegative when vaccinated, had never had a PCR positive result, and had more than 15 days follow-up after the second dose. We got UK approval in December. And the policy set by the JCVI, it was to use 2 doses of the vaccine 3 months apart. And we'd shown in our clinical trials that the antibody response after the second dose is higher when you have a longer interval between the 2 doses. We also saw very similar results in older people and those with comorbidities. We then were able to update this primary analysis, bringing in more cases, and then this was published in February, but it supported the use of the vaccine. It simply added to the data that we already had. We then moved on to vaccine effectiveness data, which was how the vaccine works in the real world. And we've seen that in England, in people over the age of 70 who'd received 1 dose from 28 days, there was at least 60% protection against symptomatic PCR positive disease. And in the over 80s, hospitalization was reduced by 80%. This is really exciting data to have in people over 70 and people over 80. The vaccine is working extremely well. And despite the different results coming out of clinical efficacy trials, which were run separately and therefore should not be compared, we're actually seeing very, very similar results from the Pfizer vaccine and the AstraZeneca vaccine in these real world effectiveness data, and also seeing that transmission is cut after even a single dose of the vaccine. Now, I just want to spend a moment on the VaxHub contributions to this project, because that was really important. In 2020, we had to design the vaccine, order the synthetic DNA we made, start making the vaccine, generate the first stock of vaccine, that's the starting material, characterize that, get it manufactured into clinical trials, and then transfer on the manufacturing process to AstraZeneca and all of their sub-licensees who are now manufacturing the vaccine. And the flexible funding that we had for vaccine manufacturing through VaxHub was what allowed us to start this process. The work we've been doing on development of next generation sequencing methods for rapid characterization of the starting material was absolutely critical to our ability to generate this starting material rapidly. In previous years, it used to take us anything up to 9 months, maybe even a year, to be certain of what we had in our starting material. And now we can do it in weeks because of the methods that we've developed through the VaxHub funding, working in our own manufacturing facility. The process development for large-scale vaccine manufacturing that was transferred to AstraZeneca was also funded through the VaxHub. So without VaxHub, we wouldn't have been able to start this project and move it nearly as quickly as we did. So now the vaccine is in use in 100 countries to date. Approvals, in more than 100 countries. It's in use in 168 countries. More than 500 million doses have been released. Around 80 million doses delivered to COVAX, and that's 96% of the total COVAX supply. Obviously, this is a vaccine that we wanted to be used widely around the world. The storage temperature means it's much easier to distribute than other vaccines, and it is being widely used around the world. And to do this, AstraZeneca have built many, many regional supply chains to make that happen. Now, what I haven't told you about in this short presentation is what hasn't been funded in the past. And for 20 years, we have struggled to raise any funding for the development of vaccine technologies. We can get funding for development of a vaccine against a specific disease, but nobody ever wanted to fund technology development, which is absolutely critical to our ability to respond to outbreaks and pandemics. We weren't funded to accelerate the initial generation of the starting material. We had plans in place to do that, to make that happen more quickly, but we didn't get the funding to do that. So we had to start from what we had done, what we knew how to do, what we thought might go better, and make our best efforts to go as quickly as we could. And that's how we were able to get into clinical trials by the 23rd of April, drawing on what we knew about the technology, but feeling at all times very hampered because we hadn't been able to do all of the work that we would have liked to have done and planned to do to make this go even faster. Had we been able to start our clinical trials earlier, it would have been very helpful. So I'm going to leave it there and just give some acknowledgements to the many, many different groups of people who have contributed to this major effort, and particularly the clinical trial volunteers, because without people coming forward to volunteer to take part in clinical trials, we wouldn't have any vaccines. So their participation is absolutely essential. I'll finish there. Thank you.
Thank you, Sarah. So I would move straight on to the Q&A. So if all the speakers from this morning, Sarah, Robin, and Steve, if you'd like to switch on your videos, please. So in this Q&A, I'm going to be supported by Marta Mauri. She's a VaxHub early career researcher, and she's going to pose some questions by the audience. I'm going to start with a question for Steve. Which links a little bit to some of the points you've made about the work of the Vaccine Task Force and what you have planned in the next few years. But we have heard also from Robin and Sarah some of the challenges that they encountered. So my question would be, what really are the priorities the next 5 years in the UK to improve the vaccine manufacturing ecosystem? And maybe linking here to one of the points made by the audience, in particular, that is interesting knowing what are we going to do about variants more generally in the UK?
I think we could take a lot of time answering this question, and I'm just going to give a couple of high level, what I think will make a big difference. I think there isn't enough big player vaccine development action in this country at the moment. So if you look at where the major hubs are, where the major vaccine suppliers of the world are doing their development, it isn't in the UK. And I think that we need to attract that kind of player to the UK. That way we will be at the forefront of technology development of vaccines that are commercially likely to be developed over the next 5-10 years. I think in parallel with that, there has to be a much, I think the way Sarah finished actually struck a chord with me about some of the things that didn't go right in the last 15 months. And I think some of the stuff that Robin's also talked about on technology and how we deploy vaccines is important for us to make sure we've got joined up ways of making sure that our infrastructure uses the investments we've made. And we've got some fantastic institutions. We've got VMIC, CPI, CGMIC, all of these players now in manufacturability coming forward. That needs joining up and a plan putting together for that 5-year period. It's totally different to the variant question. I think I'm going to let Sarah and Robin answer the variant question, because I think I could talk again about that. But actually, Sarah could perhaps talk about what she's doing, and that would be a better answer, because that's with government support too.
Okay, so just very quickly then, we have a preprint which came out, I think, yesterday, just showing a preclinical study of giving a variant vaccine either alone or after 1 or 2 doses of the original vaccine. And we see that the variant vaccine induces an immune response which is stronger to the variant, which is the beta variant, than it is to the original vaccine, and vice versa with the original vaccine. But there is some cross-reactivity with either. And then if you've had 1 or 2 doses of the original vaccine and then give a dose of the variant vaccine, you increase the immune response both to the original virus and to the variant virus. In general, the strong immune responses are cross-reactive to many of the variants. So the focus is more on getting a very strong response than a very specific response. This is going to go into clinical trials imminently. But what we don't want to do is end up trying to make multiple different variants of the vaccine and work out how to implement them. The focus is on getting a very strong immune response, which will protect well against all of the different variants.
Thank you, Sarah. May I ask Robin, I know you've typed the answer, but this can be only seen by the person who was asked the question. So, and I think this, your answer is probably going to be interesting for our audience.
So we've also been working on variants. We're not as ahead as Sarah is because her technology has already been approved and licensed. So, we're looking very much more to the future and whether you might need combinations. And I suppose one of the things that our technology offers is the ability to have, 4 or 5 variants in the same vaccine dose. And we are talking to potential partners to fund that. Although we're not currently in the phase of moving it into the clinic. We can move it into the clinic, but it's something that's there should somebody want to call on us to move forward. And I just wanted to pick up on one of the things that Steve said is, we've highlighted 2 technologies, the adenovirus technology, the RNA technology, but actually the hubs are covering a wide range of technologies, and that linkage, that coordination, I think the 2 hubs really started a very good job to link all the different key players together. But that's the beginning of the journey. I think we've created a basis, but we need to look to the future to make sure we build on what we've learned.
Certainly. There are some questions, there's quite a lot of questions from the audience, so we needed to sort of select from. So Marta, what is your, do you have any questions from the audience you'd like to ask our speakers?
Sure. So first of all, thanks for asking so many questions. Some of them have been addressed already, and I'll try to pose the more generic one to the panel. So one was about the variants, which has been addressed. And the second interesting one is whether you are both considering developing a nasal route of administration for your vaccine candidates that would protect from transmission and not only from hospitalization or severity of the symptoms.
So we already know that the intramuscular injection can have an effect on transmission and asymptomatic infection. All of that data has been published. It's less effective against transmission and asymptomatic infection than it is against symptomatic disease and then severe disease. And that's to be expected. But with the Oxford AstraZeneca vaccine, there is currently an intranasal vaccine trial being run. The difficulty is going to be going from the first studies of that vaccine to demonstrating efficacy of the vaccine. It's not possible to measure immune responses and compare them with the immune responses you get from intramuscular injection and assume that the outcome will be the same. So it is necessary to then set up a further efficacy trial, which is quite a large piece of work to be done.
Thank you, Sarah.
So we're also moving into an intranasal study with RNA. That's a really very novel approach. So nobody knows whether it will work. And I suspect that we will need to do quite a lot of optimization on the delivery for RNA technology. So that's something more likely for the future. But obviously it would be much more amenable for global distribution if we could get that to work.
Thank you. Then a second question that may concern several people is more about the mRNA or self-amplifying RNA vaccines. So actually I put together 2 questions. One is, what is the half-life of these RNAs? And the second one is the concern on whether they could affect the human genome by inserting themselves, for instance. So could you please elaborate, Robin?
So first of all, I'll take the second question first, which it can't change your genome. It can't alter your chromosome. So this is not genetic modification. And I think that's one of the advantages of RNA. RNA typically is quite labile. It stays in the cytoplasm. Now, there are differences between mRNA and self-amplifying RNA. mRNA is expressed very quickly and disappears within a matter of days. Self-amplifying RNA actually is expressed and peaks at about 7 days and can go out for up to 3 weeks. Now, we're interested in that not just for vaccines, which may mature an antibody response, but it could be very important in areas like protein replacement therapy, where you could have an RNA dose once a month, and that's something that couldn't be realized by conventional mRNA technology.
Thank you very much. And maybe I have a third question for all speakers, which is whether we should consider a non-for-profit vaccines or patent-free vaccine. What is your opinion on the topic?
So I can start off. I mean, you've heard from Sarah that AstraZeneca are doing their vaccine royalty-free during the pandemic, and I think that's a fantastic stance, and it would be lovely if the other vaccine manufacturers embraced the same philosophy. Personally, I think that although it sounds very attractive to say waive patents, I don't think it actually gets you to the end game faster, because we know that it's not just patents, it's technology transfer, and the hubs have been looking at technology transfer. That takes time. If you actually partner with somebody like AstraZeneca, you're then covered by their authorization. If you're making a biosimilar, you would have to do a large efficacy trial that would take at least another year. And so it would be a much slower approach to getting vaccines rolled out globally.
I agree with Robin, but I'd also like to add that simply waiving patents and allowing anybody who wants to try to make the same vaccine is going to be really counterproductive. We are all manufacturers are constrained by supply chain issues, and we need all of the supplies that need to be used to make vaccines to go to manufacturing sites that have the technology up and running. AstraZeneca has more than 20 of them. Other manufacturers are looking to expand, where the technology not only to manufacture the vaccine, but to test the vaccine, to do all the rest of necessary quality control and release it in a way that's approvable by the regulators. That's where those resources have to go to. And by making the IP freely available, that doesn't mean that you can make the same vaccine.
And if I could throw my hat in here as also just say that I think one of the other pieces that just what Sarah just said is really important. As you look forward 10 years, what Robin was talking about having a very portable vaccine manufacturing facility is very different. But again, it's not waiving IP, because you'd still need to have people who are trained and able to go with that and operate it and then be able to release a vaccine. It's one of the things I think this crisis has probably taught lots of people in the business is how important the release of the vaccine is at the end of the process. It's not just the authorization. It's the ability to be able to say that vaccine is now usable by the local population. And that's really critical in any of this that we do going forward.
Perfect. Thank you very much. And I think we may have time for one more question, or should I conclude here, Martina?
In the interest of time, I'd like to thank the speakers. I know there are lots of interesting questions in the chat. If I can remind people to use the Q&A chat, I know this is confusing. Zoom has a chat and then the similar icon for a Q&A chat. If you could use the Q&A, that would support us in managing the question. And maybe some of the speakers can also view the question and answer, not live, but answer in the Q&A chat in case there is no time to handle those live. So thank you to all the speakers, and I'm going to pass on. Thank you, Marta, and I'm going to pass on to Ben for the hub introduction.
Great, thank you, Martina. And from my side, thank you to all of the speakers from session one. It was a really instructive forum and exciting discussion during the panel Q&A. So my name is Ben Pierce, Operations Manager for the Imperial Hub, and I'll be chairing session 2, focusing on the UKVN vaccine manufacturing research hubs and our roles in global manufacturing capacity building. I will be assisted by co-chair Paul Katenwe, our early career researcher based in Uganda. And may I ask all the speakers to be promptly ready to take control of slides, and we'll need to be a bit strict on keeping to time because we are now running over. So I will begin by giving a very brief summary of our activities. And I will do my very best to include as many exemplars as I can, and apologies to those that I just could not fit in for the interest of time. So our hub, and this is one hub based at Imperial, it's a global organization. It's mostly based in the UK. On the left of your screen, you will see UK-based research institutes, and you can read the logos yourselves. There's NHS Blood and Transplant. At King's College is a new member, previously Cranfield University. And I'd like to highlight the inclusion of Center of Process Innovation and National Institute of Biological Standards and Care. And all of these universities and research institutes or centers link through 8 work streams with vaccine manufacturers in developing countries, whether that's MI Hisson in Dalian, China, Hillman Labs in India, Incepta in Bangladesh, UVRI, the Uganda Viral Research Institute in Uganda, for biotech in Vietnam, GSK, but primarily the GSK Vaccines for Global Health, GVGH, based in Siena, and Developing Countries Vaccine Manufacturing Network, or DCVMN, a consortium of 42 member companies located in developing countries. Very proud to have expanded our reach and breadth and depth in just a very brief time, I mean, less than 3 years of activities, to introduce and welcome new members, University of Leeds, BioE in India, Minhai from China, and UK SMEs, MBio and Excivian, who have linked with CPI and NIBSC to expand and inform further on their vaccine candidates against Zika and pneumococcal disease, and NSC, a company that's linking with Robin Shattuck to look towards a solid-based implant for siRNA delivery. The need for the hub was quite clear in 2017, but the reasons were different. As Steve mentioned, the world has changed. We now have other additional reasons for more improved and further vaccine manufacturing research, but then it was based on population growth mostly, that there's a need for more vaccines and better manufacturing and further high quality technologies and innovation to be injected in the manufacturing pipelines that are directed to impact ODA, official development assistance, or developing countries, simply based on population growth. More people means more doses are needed. However, as we've all experienced quite tragically, the world has been turned upside down. There are other reasons now, and it's not only health-based, but economic-based as well. And simple investment in research at earlier stages can drastically, can significantly avoid economic shakeups to the global economy. And here's just one bit of data that I've taken from the BBC. Of course, you can find other examples as well. That's in addition to the horrible toll that we've seen in terms of health that's affected most of us on some personal level. But the way, pardon me, the way we set up the hub was a bit different. Rather than focusing on pathogens of interest, we worked on platform technologies, and Robin touched on this in one of his responses, that really our activities are based on 4 pillars of research, and they're each shown here. One is generalized modules for membrane antigens, or GEMMA for short. This is led from the GVGH GSK group from Siena, Francesca Micoli and Elena Palmieri, and this derives from gram-negative bacteria that produce outer membrane vesicles that are engineered to upscale and produce further amounts of these vesicles. And these are really powerful technology and highly mature technology that have been further developed through links with the NIBSC and Imperial College as well. Now, you will hear more about the second and third technology shown here from talks following Martinez after me, which is baculovirus. This is led by Professor Emery Berger from the University of Bristol, and you'll hear from Dr. Do-Tuan Dat from Biotech, who's linked with Professor Berger on collaborations and tech transfer secondments and looking to take up this innovative technology and new way of making vaccines in Vietnam. Yeast are a traditional platform of creating vaccines, but we have innovative strains as well of interest here, and you will hear from Manuel Ahassan, a researcher lead from Incepta in Bangladesh, and how that company has learned of tech transfer and received training from researchers at Imperial College. We're also looking at other strains of yeast, Saccharomyces from Jianing Zhu, and others in the hub to help the development of new vaccines against other pathogens. And we've all now know much more about RNA from the last year, but of course, as recent as today with Robin's talk. But what I think I'd like to emphasize here is the strong links within the hub between Robin's team and institutions like GSK, NHSBT, NIBSC, CPI, and the Uganda Viral Institute as well, towards pushing this technology further. Now, a common thread throughout is our use of quality by design. This involves our Department of Chemical Bioengineering, Nilay Shah, Cleo Kantaravni, a really strong early career researcher, Zoltan Kiss, are just a few that I would like to point out. Modeling is used to help support this, and regulatory alignment with feedback and input support from NIBSC. In addition to that, all of these vaccine technologies are supported by really exciting research activities in delivery systems, whether that be new polymer systems from Professor Cameron Alexander, Pratik Gurnani from the University of Nottingham, polymer-based systems from Professor Molly Stevens' group at Imperial, or vesicle-based systems from Rongjun Chin and his group. And these are all supported by other kinds of carrier technologies, such as ionic liquids from Professor Jason Hallett's group at Imperial, with support from early career researcher Talia Shmuel. And I'll make one exemplar from that team in just a couple of slides. So with these exciting technologies, we are at liberty to choose amongst dozens of pathogens. And in 2017, a lot of these decisions were based on not only priority pathogens as determined by the UKVN, but the number of epidemics that have arisen in the preceding years. And here's one example based on data collected by the Wellcome Trust, and a number of these pathogens are focused on by the hub, and that has expanded in less than 3 years' time. Shown here are pathogens of interest in some way, and some very heavily as core streams of activity. And very excitingly, through careful and close change management, we were able to take on SARS-CoV-2 within an extremely excitingly rapid amount of time. And that's under the experiences of lockdowns in institutions located in China that saw the first national lockdown, in northern Italy that saw the second major national lockdown, and the UK. And so thankful to all hub members that continued and remained, they persevered through a very stressful time for themselves and the rest of the world. And through this experience, we're very proud to show off our training. Now, this includes secondments of international staff members from vaccine manufacturers in Uganda, Bangladesh, and China to labs at Imperial College London and Vietnam, and staff members from Vietnam to the University of Bristol. Very kind contributions from companies such as Dash in partnership with Brompton Bikes to donate foldable bikes for some of our team members in the Shattuck team. And through these efforts and this team, an open collaborative based relationship, very proud to announce that we recently were awarded the Imperial President's Award for Outstanding Research Team. So well done to all of the lab-based researchers, particularly for that. Very proud to have alumni coming out from the hub and take on independent research opportunities, but also training workshops that have been received by developing vaccine manufacturers based in developing countries. I was blessed to be able to join and organize a quality by design workshop delivered in Hanoi, Vietnam in November, just before the first outbreaks of COVID-19. That was attended by over 50 DCVMN member company staff, and really exciting outcomes to them. And it was exciting to see the eagerness of these companies to take on innovative ways of using quality by design. And well done to Zoltan Kiss from our hub and Maria Papathanasiou from the VaxHub as well. It was one of our first joint efforts. Early career researchers Rochelle Fru-Rabe and Parisa Masami, co-chair of the next session, delivered a really nice innovative strain yeast-based and baculovirus-based manufacturing workshop to DCVMN members during the lockdowns in May 2020. So that was one of the first virtual events to come from that. And we are planning other events and activities are ongoing, including RNA vaccine training program planned in Uganda in September 2021. We are just as proud as our outputs, and this is a selected list. This is absolutely not exhaustive. I'm pleased perhaps from the recording you're able to find more information at your leisure by looking at these DOIs, a range of technologies being described here, and it's not only based on vaccine technologies, but one example here is the identification of linoleic acid as a binding factor within the glycoprotein pocket of the SARS-CoV-2 antigen. And that's from Professor Emery Berger's group with substantial support from our hub. Robin Shattuck has been on the media. I hear my boss's voice late in the evening sometimes. And so thanks to him for all of his media engagement and that of all vaccine researchers and spokespeople from the hubs and some that are in attendance today. We have been quite active in policy engagement. We have visited and met with international members from policy forum meetings or personal visits from the Chinese Science and Technology Minister in July 2019. You can find multiple examples of our research online, whether that is a case study from the Center for Process Innovation, CPI, and their assistance and support for new ways of manufacturing virus-like particle polio vaccines, or highlights of Jason Hallett's technology, for example, use of ionic liquids that can maintain stable saRNA delivery within at least 50 days at room temperature. Highlights of our scientists engaging with the public and encouraging vaccine uptake. Highlights from hub researchers such as Rongjun Chen and Zoltan Kiss on the details of how China is able to manufacture vaccine to carry out their very impressive vaccine programs. Apologies, this is cut off, but a highlight of a scientist, Kai Hu, in Robin Shattuck's lab on that, him being from Wuhan and being a key integral part on researching and developing a COVID-19 vaccine candidate, amongst other examples, whilst maintaining strong and excellent policies within our hub to support mental health measures and safeguarding, as well as promoted by the EBSOC. So with that, I'd like to hand over the control of the slides to Martina from the VaxHub.
Thank you, Ben. So I'll try to be as quick as I can to leave as much time as we can to our speakers. So running the hub has been an incredible journey. Just taught me a lot about different working cultures, but also it's quite a large and complex system to run, like Ben highlighted. Our hub supports a program of research focusing on all aspects of vaccine manufacturing of benefit to developing countries. So ensuring the supply, translating new technologies to manufacturers, and offering rapid response technologies in support of epidemic threat scenarios. The hub essentially funds on-site researchers working on three main technologies: VLPs, viral vector, and conjugate vaccines, and platform operations, where essentially the hub acts as a funding body to advertise, select, and award projects by competition. In our hub, UCL and Oxford are the two main hubs working together with different spokes, Imperial, Leeds University, and the London School for Hygiene and Tropical Medicine. But we have interacted with different stakeholders, and we have a group of members, see them at the bottom, who routinely participate and engage with us, steering the research direction priorities. So this slide shows in numbers really the size and some of the outcomes that we have achieved as a team in the last 3 years, despite some of the university closures, because of the fact that the hub really brings a critical mass of researchers working in a specific area. It attracts more funding from different sources, so UK and abroad, so you see the figures is particularly high, and we have running a number of different associated projects. I'm now going to provide just a few more detail for some of these categories. So it's important to say that our hub focus is not really to develop a vaccine against a specific pathogen, but to propose different vaccine technologies, but also different engineering approaches, potentially applicable to multiple vaccine development routes. So our work packages are very varied, focus on solutions to speed up process development, to supporting scalability, development of assays, new approaches to formulations, but also computational tools, really with a high level of interactions between different partner institutions. And you'll see some of the outcomes at the right-hand side. In my next slide, I'm going to just show selected examples of projects we have funded. So some focus on what technologies in particular, like the work from Leeds on a polio candidate. Other aimed really to apply some novel methods, but applicable to all technologies. And we had informative technical webinars for our developing countries partners where they could engage with all the proposed solutions. Some of these projects are ongoing. We are organizing actually a webinar series from all of these in fall. In terms of our training effort, so this was an important part of the hub's remit. Our aim in this space was twofold. So we supported places from our partners to participate in different courses offered in the UK, in US, in Africa, which also provided excellent networking opportunities. But we also developed a program for our on-site researchers in addition to the technical program. So for example, some examples here on GMP, policy engagement, media engagement, obviously given the number of requests and interest that we had from the press in the last few months. So, and the aim is to really repurpose some of these. We were planning one in Asia, so maybe we can do that by before the app ends. And finally, we developed a number. We developed a number of activities to really engage our users group, like for example, webinar series, monthly newsletter, but also policy engagement activities. Some of these are led by our policy advisor, Penny Carmichael, who is running this seminar today, and including participation to UN and WHO initiatives, one of which is led by our next speaker, Jisui Dong, and we've published different media outreach articles and podcasts. We're planning a few more, so join our Twitter and watch this space. I'm going to hand over to Ben again for the next session.
Great, thank you, Martina. If we can hand control to Jisui Dong, the unit head for the WHO Local Production and Assistance Unit, to provide holistic, strategic, and coordinated support to member states in strengthening sustainable local production of safe and effective medicines, vaccines, diagnostics, and other health products. Jisui, over to you.
Yeah, let me try. Good morning, everyone. Thank you, Ben. Let me try, I can move my slide. I think it seems I have a little problem to move my slide. You are controlling.
Jisui, I can do this for you. Just say next slide.
Okay, sorry for that. Thank you, Penny. So my topic is a holistic approach to local vaccine production, key success factors, and the role of universities as enabling partner. So the COVID-19 pandemic represents an unprecedented global health challenge. In combating the pandemic, a lot has been achieved, but most needs to be done. COVID-19 vaccine has been developed at a groundbreaking speed. Today, 8 COVID-19 vaccines are listed by WHO for emergency use. As of 2nd June this year, over 1.6 billion vaccine doses has been administered globally. But more doses are needed, and they are challenged to overcome to meet this need. So what we learned from COVID-19 pandemic? You can see first, we said we can leverage the existing scientific knowledge and the research to speed up the discovery of the solutions to combat the virus. Research in the coronavirus puts us helped to identify the spike protein sooner. Second, a focused global effort helped to reduce the time needed to develop and produce COVID-19 vaccine. Many stakeholders, including government, industries, the scientific and international communities, regulators, prioritized their effort to find effective vaccines and other tools to combat the COVID-19 pandemic. Third, the collaboration and cooperation within the global community is key to facilitating the global effort. The gene sequence was shared, resources were mobilized, global initiatives such as ACT accelerators and the COVAX facility were established. Within the industry alone, over 270 partnerships were formed, of which 214 were related to COVID-19 vaccine production. Collaborations in the short and the medium term aim to increase COVID-19 vaccine production capacity and ensure equitable distribution of vaccine globally. In view of pandemic preparedness, long-term collaboration are essential to ensure that sufficient capacity is both generated and retained. Therefore, commercial viability is critical, absolutely critical, to ensure that this capacity is utilized on an ongoing basis and not lost. Next slide, please. So as we know, vaccine manufacturing is complex. There are key elements for the sustainable production of a quality, safe, and effective vaccines. It is essential that a government have a long-term commitment to support local production by citing coherent policies, providing time-bound incentives, and building an enabling business ecosystem. A strong regulatory system is important to ensure quality, safety, and efficacy, and also enable timely market entry. Access to skilled workforce is critical, especially for vaccine production. However, the lack of local expertise is a continuing challenge for low and middle income countries. Of course, partnership and collaborations are also essential to local vaccine production. For example, with over 100 components in vaccine production, partnerships with raw material suppliers are important in securing the supply. If one component does not come in time, production could be stopped and the batch may need to be discarded. Next slide, please. So what is the role of universities and research programs play in combating COVID pandemic and also in local production of vaccines? We say university and research programs contribute to local production of vaccines in many ways. I just listed 3 here. First, universities train people. Skills development and provision of qualified talents are vital for vaccine development and production, especially in low middle income countries. Universities with lab or pilot scale facilities could provide hand-on training to reinforce technical skills in manufacturing and quality control testing. Second, universities and research programs can drive innovation for new vaccines to treat new diseases, new vaccine formulations to suit the local context, such as for improved product stability outside of the cold chain, and for new manufacturing processes or technologies. For example, continuous production system to increase production output and at the same time maintain a small footprint, operational footprint. Given the strength of universities in discovery and innovation, they can support effective translation and adoption of new technologies by industry. Last but not least, universities can play a role in vaccine manufacturing using a hub and spoke model, with the university as a hub. Engaging universities on a continuous basis in the vaccine ecosystem could reduce the time frame to address future pandemics, as the partnerships and the shared knowledge within the ecosystems are already in place. As seen in the current COVID-19 pandemic, collaborating with universities and research programs could help speed the development of products to tackle future pandemics. Next slide, please. So this slide shows what WHO do in supporting member states to strengthen local production of vaccine and other health products to achieve quality and sustainability. So we nurture strategic partnership with member states and different stakeholders, including universities and research programs. We conduct feasibility assessments to identify gaps and propose recommendations for sustainable local production. We support member states to develop and implement their national strategies and roadmap to promote local production. We also provide, very importantly, needs-based capacity building and technical assistance. This May, a resolution on strengthening local production was adopted by the 74th World Health Assembly. More than 100 member states co-sponsored this resolution, which showed great interest and commitment in promoting local production by member states. There is also new WHO initiative we called the World Local Production Forum, WLPF in short. This forum will be regularly to provide the global community a global platform for open dialogue and collaboration and addressing key challenges in promoting local production and technology transfer to improve access and strengthening health security. We are currently convening the first WLPF, which will take place from 21st to 25th this month. You are all welcome to attend. So next slide will show some examples of our work. I said we provide a lot of tailored-based training and technical assistance. Last year, we organized the first virtual CGMP training marathon from September to November. It lasts 3 months, 12 weeks. Every week, there were 1 topic. So over 1,000 participants around the world attended each topic weekly. And this year, our training will focus on GMP in vaccine manufacturing, facility designs, and technology transfers. You can see on the right hand of the screen, there is a holistic strategy setting, which we supported the member state, like Ethiopia, in the developing and implementing their national strategy and the roadmap in promoting local production. So a lot of progress has been seen in the countries, and we are still working with the government and to implement this strategy as planned and also adapt the changing business environment. So with that, I thank you all. Over to you.
Thank you, Josue. What a lovely presentation, and thank you for all of your contributions. I'd like to pass over to Dr. Neni Nureni, Research and Development Project Senior Integration Manager for PT Bio-Farma in Indonesia. Dr. Nureni was also part of the Research and Innovation COVID-19 Task Force under the Ministry of Research and Technology of the Republic of Indonesia. So Dr. Nureni, off to you.
Thank you, Ben, for the nice introduction, and thank you for the invitation. So today I would like to share our experience on the capacity building in the vaccine development through successful partnership with the VaxHub. I cannot, yes, okay. So let me introduce our company in brief. So Bio-Farma is established 130 years ago. So we moved to Bandung. Bandung is located in West Java, Indonesia, in here. And we are state-owned enterprise, 100%. We produce the vaccine and anti-sera manufacture. And also we implement all the quality integrated management system. So our vaccine has listed as a WHO pre-qualified vaccines with the capacity about 3 billion doses per year, we can supply our vaccine to more than 150 countries in the world. So this is the list of the vaccine product from biopharma. Sorry. That already got the WHO PQ. Sorry for that. So I think maybe Penny, can you help me to handle the slide? Thank you.
Yeah, which slide would you like to go back to?
Back, yes, back.
This one. Okay, just tell me next slide when you want to move forward.
Back, Penny. Thank you. Go back, yes. Back, yes, this is okay. So, yeah, next slide. Okay, so this is the list of the biopharma vaccine product that has all the WHO pre-qualified vaccine. So there's 14 antigens, and then we also export our API or active pharmaceutical ingredients also to the other manufacturer in Asia. And recently we got the WHO emergency use listing for the novel oral polio vaccine type 2. Next slide, please. So this is the list of existing acquired vaccine technology platform that we got. So live attenuated vaccine, inactive vaccine, subunit toxoid, subunit protein, and subunit recombinant protein, and also polysaccharide conjugate vaccine. Next slide, please. So this is the strategy for the COVID-19 vaccine supply in Indonesia. So we have 3 strategies. In short term, we focus on the immediate supply. So what we have is to do the technology transfer, formulation, and filling, utilizing COVID-19 vaccine bulk from the candidate partner. So in this case, we have collaboration with the Sinovac China, and the vaccine is already got the emergency use authorization in Indonesia and deployed for the vaccine program in our population. In the midterm, our intention is to have the self-reliance in the vaccine, starting from the upstream until downstream process. So this is about the national capacity building. So we are together synergy with the ministries and institutions in Indonesia to develop the recombinant subunit protein. In the mid-long term, we would like to acquire new platform technology for the vaccine production, and in collaboration with the VaxHub, WHO, and also CEPI and other international institutions. Next slide, please. So this is the acquired new platform technology that we have, and then the pandemic is changed our landscape of the vaccine development. So we need the rapid response platform technology. That's why biopharma very interested to collaborate with the VaxHub and other international institutions to gain the capacity building on production of the viral vector or RNA platform technology. Next slide. So we see that the VaxHub is very important as an access to technology transfer, capacity building for improving response to future epidemic and pandemic. So our request is to access know-how from the vaccine hub, comprising of the vaccine seed and design, production process, QC methodology, and formulation technology. And of course, the capacity building to get the training, to acquire expertise in rapid response technology, development of the vaccines against difficult pathogens and robust delivery system. Also regulatory oversight on the platform of the pandemic vaccine and support on the clinical trial activities. Next slide, please. So partnership with VaxHub, we see that it is very important for our researcher to increase the professional development by either by the PhD program. So we have the biopharma staff in doctoral program in UCL and Oxford University, and also by a training course for the industry. For example, join the vaccine MBI course, the vaccinology course, the vision leadership, and also the process development and analytic webinar series. Next slide, please. So it is with the support from the VaxHub, we got funding with the UCL for the VaxHub feasibility study on the optimization of upstream process for the recombinant COVID-19 expressed in Pichia pastoris. So general aim is to build the capacity of Indonesia to develop the SARS-CoV vaccine based on the recombinant protein. So this feasibility study conduct from November last year until July this year. So the specific aim is to construct the vaccine antigens, develop it by process, and evaluate the quality of the antigens. Next slide, please. So this is the, as you can see here, this is our target antigens is the receptor binding domain on the spike protein. So for the clone preparation and clone development, we collaborate with the University of Pajajaran in Bandung, one of the university in Indonesia. And then this clone has already sent to the UCL for the further development. So for our status right now is in the clone expression and the optimization of the upstream process using the design of experiment methodology and also clone characterizations. So once the parameter is established, we will implement this parameter to produce the vaccine antigens in the setting of the industry. Next slide, please. So next step of our feasibility study is to continue the upstream optimization together with UCL and biopharma, and then downstream optimization and quality control development with the also Viranova, and a preliminary preclinical that we would like to do the immunogenicity study in mice model that we'll conduct in a biopharma. Next slide, please. So the collaboration with UCL, not only for the COVID-19, but also for dengue vaccine production, because the dengue also a major health problem in Indonesia. So our project goals is to produce the recombinant VLP in Pichia pastoris, and achievement to date is we got the new vaccine prototype, adaptation of developed protocol, and also USP process optimizations in progress. Next slide, please. So with this, I would like to thanks to all the member of the COVID-19 research team in biopharma, UCL research team, Salome and Eli, and VaxHub team that really supportive in our collaborations. And thank you for your attention. Back to you, Ben.
Thank you, Nani. A wonderful presentation and great example of collaborative efforts between your team at BioPharma and UCL's VaxHub. Thank you very much. It's my pleasure to introduce the next speaker, Mohammed Mano Ahsan from Bangladesh, from Incepta in Bangladesh. Mano is head of research at Incepta, the largest vaccine manufacturer in Bangladesh. He's been supervising several vaccine process development initiatives, including rabies, rotavirus, cholera, and a number of other pathogens within Incepta's work streams. So Mano, if I can hand over the control to you.
Yeah, thank you, Ben. Thanks for the introduction. So basically, it's my pleasure to present in front of you. Actually, yeah, it's working now. So at first, I would like to say at a glance about the Incepta. Basically, Incepta was established in the year of 1999 as an Incepta Pharmaceutical Limited. And after the inception of 5 years of inception, we became the second largest company in Bangladesh. At present, we are producing 1,100 products for local and industrial market. And at the moment, we are exporting our products to 74 countries, including United Kingdom. So the company started its vaccine business in 2011. We are the first and leading vaccine manufacturing company in Bangladesh, and it is we have the state-of-the-art facility fully compliant with full GMP requirement. So I would like to say something about the infrastructure and capacity. We have a production for the bulk and seal production. We have a 5 liter to 500 liter fermentation capacity with all supporting downstream processing. Recently, we also installed a fixed bed bioreactor system from Universal. So this facility is capable of to produce recombinant bacterial and viral vaccine. And our fill finish capacity, we have 4 lines with capacity to fill around 1 billion doses yearly. If you consider in multiple format, we can use either vial, ampoule, refill series, or lyophilized product. And for quality control, we have a dedicated lab for analytical and microbiology. Also, we have a dedicated animal house for in vivo testing, and it is probably the largest animal house in the country. We have a dedicated R&D lab. The R&D lab is equipped with modern fermenters up to 990 liters also, and fixed bed bioreactor along with downstream processing equipment. We have a dedicated area for handling virus and bacteria, and also for recombinant work. Can I give the previous slide? Yeah. So initially we started the business with the fill finish product. So we have 8 vaccine and 5 immunoglobulins in the local market. But in parallel, in the year of 2012, we started our own R&D. So from the R&D, so far we have developed 5 products, which are in detail. For our first product, the Ingovax SYW, which is basically meningococcal polysaccharide vaccine. This vaccine we obtained the license in Bangladesh, which is already now in the Bangladesh market. Then and second one is hepatitis B. This is basically yeast BNP-based vaccine produced in yeast, but the clone actually we haven't developed the clone. We procured the clone from third party, but the fermentation and purification we developed. So this vaccine is also got the license in Bangladesh. Our third one is cholera vaccine. We also got the license in Bangladesh. So our rabies vaccine, which is developed in SCLS platform from Universal Belgium, we recently just finished the clinical trial, which is about to get the licensure in quarter 3 of this year. Our typhoid conjugate vaccine, which will enter the clinical in the quarter 3 of this year, and our meningococcal conjugate vaccine, which will enter the preclinical in quarter 3. So in addition to this, we have many more products in the R&D development stage, mainly human papillomavirus vaccine, pneumococcal polysaccharide vaccine, measles, rubella, and also CRM197, which is carrier protein. And also now for sustainability, and also we are now focusing on the platform technology development, targeting the SARS-CoV-2, mainly mRNA and protein subunit vaccine. So, though we had a product hepatitis B based, and I told you earlier that we didn't develop the clone. So we wanted to develop a yeast-based platform technology at Inceptor. That's why we actually collaborated with the Imperial Hub, and we started the training in 2019 in the Dr. Karen College Lab, Department of Chemical Engineering. So our primary target was to develop a L1 human papillomavirus L1 based DLP for all 6, 11, 16, and 18 serotypes. Also to develop a CRM197 protein, which is basically a carrier protein used in glycoconjugate vaccine. So during this training, we utilized actually we tried to develop the yeast expression of which can secrete the protein into a supernatant. So we utilize the IP3 plasmid, which is and which has got a modified alpha factor for secretion, and also using the wild type yeast strain. So during those training, we developed 4 ways express all 4 types of the HPV. One for expression, and which is produced actually now We got the clone which is expressed, also CRM1 and 7. So all the 5 clone, we got the material from the Imperial in 2019 in October. So after receiving this clone, so at Incepta, we developed a fermentation and purification process at 10 liter scale. If you have a look here for HPV VLP for 18, under the board reducing condition, the HPV L1, you can see in the one of band which is appear around 250 kV. That suggests that this is not the VLP. And if you have a look at the size expression HPLC, we compared with the reference BLP, and you can see the reference BLP retention time and the hours on is different. So from this period around, we assume that the L1 protein does not self-assemble into VLP. Instead, it expresses pentamers. Though here electron microscope data is messy. So this is for HPV18. We got the signal result for other 3 types. In this context, we have decided to offer intercellular expression in yeast. So for intercellular expression, basically now we are utilizing a commercially available platform, PCA-Pink platform, which is basically So we are trying to express the protein intercellularly. So at the moment we are cloning the codon of L1 protein into the vector. So basically, now we are utilizing what we have been learning from the Imperial course. Now we are implementing at Incepta. So now this work also supported by the Imperial Hub. So now basically I will move to our second project, CRM197. So after receiving the clone, we developed the fermentation and purification at 10 liter scale. And you can see after purification, we got a very nice and pure product. And we compared our product with the commercially available product in HPLC and found the retention time almost same. That means it has the same tridimensional structure. So after I told you, this CRM197 basically is used in like a conjugate vaccine in conjugation. So then we try to whether the each direct CRM197 can be used in conjugation. To answer this question, basically, we developed a typhoid typhoid conjugate vaccine with the each derived CRM, and we also assess the immunogenicity in mice. If you have a look after the first dose, the unconjugated VI and conjugation VI, you can see there is a difference of the response and the VI IgG response, which is more about 5 times more compared to if you compare to the unconjugated one. But after giving the second dose, booster dose, you can see there's a difference, and also there is a booster difference, which is basically benchmark of a protective typhoid conjugate vaccine. So this is here. I would like to mention one thing. This is the first report. The expression of CRM197 protein in yeast, which was previously anecdotally suggested not to be possible. So basically, this work is unique. We already submitted a manuscript to the journal for publication. So basically, this is the update from our actually Inceptor and Imperial Hub Director project. So here actually, I would like to thank Department of Health and Social Care for their funding. I hope they will keep continue this type of funding for in future. Also, I would like to thank Imperial College team, especially Robin Shaddock, Karen, Rochelle, and Ben. Finally, I would like to thank Professor Nicola Esterhaus at University of Leeds. Thank you very much for listening.
Thank you, Manuel. A wonderful presentation and a great example of the exciting promise of Pichia pastoris following Nenny's presentation as well. From my side, a massive thank you to Karen Polizzi and Rochelle all for their excellent collaborative support. I'd like to hand over for the next presentation, which is Dr. Do Tuan Dat, President of the Company for Vaccine and Biological Production No. 1, or VBiotech, a state-owned company belonging to the Ministry of Health of Vietnam. VBiotech has produced several vaccines such as recombinant hepatitis B, cell culture-based hepatitis A, inactivated Japanese encephalitis, and oral cholera vaccines, and another collaborator, core member within the Imperial Hub. Please, Dr. Dat.
Okay, thank you, Ben. And good afternoon here in Vietnam. And today we want to talk about that kind of baculovirus expression system. It's new platform for pandemic vaccine development transferred to Vietnam. And it's that kind of support from the kind of FVMR Hub. And right now we have some kind of soft introduction about our company. Is that Biotech is one company, is state-owned company belong to Minister of Health of Vietnam. We are kind of leading company in Vietnam for manufacturing, trading, researching, and developing a wide range of vaccine and biological for human use. We are kind of biggest vaccine manufacturer and supplier for our EPI in Vietnam. Our health, the kind of fourth key product, commercialized product right now is that hepatitis A vaccine, orange cholera vaccine, Japanese encephalitis vaccine, and kind of hepatitis B vaccine. On kind of vaccine, we have followed that kind of inactivated vaccine, and also is that kind of recombinant vaccines. We also have some kind of activity like R&D activity to develop our own vaccine and commercialize in our country, like kind of our commercialized vaccine, and also develops a new vaccine based on the cell-based vaccine, like the rabies vaccine and also the influenza vaccine. We know that the kind of challenges for pandemic vaccine development right now is that the kind of we could not predict the nature of the emerging pathogens and time for vaccine development, and also for costs associated with vaccine development and production, and how the kind of production capacity, and also for how the kind of global distribution and vaccination. It's for that kind of developing country, it's that like the COVID vaccine, is that a question for us, is that how the kind of technologies that kind of developing country can go for the kind of vaccine development. Right now, like COVID vaccine, we have the different kind of platform, like the live attenuated, inactivated, viral vector, protein-based vaccine, and nucleic acid vaccine, like DNA and mRNA vaccine. Is that one the kind of very challenge for the pandemic vaccine in the developing country is how we can go for large scale production. It's like the how we can have the kind of GMP facility, have the know-how method, and the kind of productivity high in, and also we talk about that kind of sorted resource. We can have like material, have equipment, and also supply chain or something like that. And it's so important one is new platform and technology, how the kind of manufacturer can reach to develop a vaccine. We have to follow the kind of collaboration with some university in UK for the FVMR Hub, like the Imperial College London, and also the kind of Bristol University. We could go for the new platform for baculovirus system. How we can choose that kind of system for vaccine development is some advantages we can have, like the kind of serum-free suspension culture, and how we can have the kind of effectively baculovirus vector construction. And also we have the high-yield culture, and we can easy to increase and improve for industrial scale applicability. And that kind of vaccine platform is safe because it's non-infectious and non-replicated vaccine. Our reason, we already have the kind of expressed ours is that kind of spike protein. It could spike protein on that kind of platform based on baculovirus vector. And we also developed the kind of production process from the beginning, like the kind of cell bank, like the kind of seed virus, and also go for the kind of all the upstream and downstream process, and also formulation. We have the some reason we go for big data and like some kind of animal like a mice is the hamster for challenges, and also is that for monkey. Here we can see some reason we have for that kind of mice is that like the PINT titer for that kind of dosage. Right now we use like 10 microgram of S to protein. Is that very high, that kind of PI, is that the high titer of the NGS, high titer PINT, and also follow the day, so 46. And also is that the kind of cellular immunity, or also good and compare the own more focus on TX1 like the IF and alpha, and also we have low the focus on TX2 like the EN4. We can see for humoral and cellular immunity for that kind of bacterial viral vector vaccine. We think that the plan for the near future right now, we can develop the kind of new COVID vaccine, again, a variant based on the bacillus BVES, the platform. We think that the kind of project, we also get the new support from the FVMR Hub, and we can have the kind of quick data from preclinical data should be coming soon before we can end on the September this year. And also we can move to next clinical trial from the accomplish on the kind of preclinical data for our vaccine candidate as a booster and variant vaccine. And also we can, based on that our platform, you can develop the new vaccine like flu, rabies, and other pandemic vaccine based on that kind of new platform. And that we can approach to new platform for pandemic vaccine development, such as other viral vector and nucleic acid platform also. And the lesson we should learn from that collaboration is that how faster the vaccine manufacturer from developing country could approach to new modern technology, and how the quick kind of vaccine manufacturer from developing country could be applied new platform for cell production for pandemic vaccine in their country with probably varied scale and large scale. Okay, we have some acknowledgement to Imperial College London and FVMR Hub like Robin Satot, Benjamin Pierce, and also is that University of the Bristol and Dr. Professor Amberberger, Rabbi and Parisa, and also that kind of group from biotech, they can come to UK to learn more about the technology and also apply good technology to Vietnam, like Mr. Chong and Tian, and many thanks for all of you. Thank you for your attention. Over to you, Ben.
Thank you, Dr. Dat, and thank you to all the speakers of today's session 2. I'd like to introduce you to 2 other hub members that will join as panelists for the Q&A session. There's Rochelle All, an early career researcher in Karen Polizzi's lab at Imperial College London and part of the Imperial Hub. And welcome to Professor Ellie Kashavarts-Moore, Professor of Bioprocess Science and Enterprise from the Department of Biochemical Engineering and part of the VaxHub. We've seen an example of her collaborative work within the VaxHub from Dr. Nani Noorani's presentation earlier in the session. So for the speakers and panelists, please do unmute your microphones. Feel free to turn on your video as well. I would first like to ask a question. This was selected with the help of Paul Katenwe from the Uganda Viral Research Institute in Uganda. So thank you, Paul. Rochelle, we've seen some of your collaborations within the Imperial Hub in working with researchers from Incepta, and it'd be really great to hear your response to a question from Stephen Morris. Could you comment on the role of academic institutions to provide training in vaccine development and manufacturing to provide talented personnel needed for the expansion, development, and manufacturing capacity in the future? Rochelle?
Yep, thanks, Ben. I think it's like we've heard a lot of examples right now of how important the collaborations are in terms of driving forward not only novel techniques that might not be available in these low to middle income countries, but also taking techniques that we have and technology that we have and transferring them to manufacturing capabilities, which we might not have as an academic institution. So the advantages come from both sides of it, from obviously like from our exposure to it, working with Manual in sector, you know, we've managed to get publications out this. Some of their staff came over and trained with us for several months, and they have now been able to take that information back with them so that they themselves can lead this technology on further. Also, as an early career researcher, this has been great exposure for me. I have, like you've mentioned previously, been involved in the DCVMN workshop with some other researchers from Bristol University, where we had the opportunity to train vaccine manufacturers from all over the world on different aspects of it. And I feel that it's really important to look at it from a broader picture. There's been a lot of conversations about disconnect between academia and industry. And, you know, as an early career researcher, I've gained a lot of information on how this all fits in together and potential roles to take that forward in the future as well.
Thank you, Rochelle. Thanks for your response. It's really an interesting response. Turning to Professor Keshavarz-Moore, could you please let us know your response to how is connecting with vaccine manufacturers supported manufacturing capacity on the other side, that is within the UK?
I think this is an interesting question because vaccine has been something which has not been in the public eye because of all the diseases which have been vaccinated for, apart from the ones that we give to our very young children, have been in other parts of the world. And all of a sudden in the UK, with this pandemic, which has been unfortunately affected a lot of people globally, maybe it's got a sort of a silver lining, and the silver lining of it is that we in the UK and other countries have become very much aware that this is not just a localized problem, but it's a global problem. UK has always been very big with vaccines, if we go back to the times of Jenner, if I could say that. So we really have it in our DNA in this country to be able to work with things like that. And of course, that was quite big during when Wellcome was a company in its own right. And somehow it sort of petered off a bit. But now it is obvious that this is important to deal with it. But one thing which has been particularly interesting is the understanding of the importance of having manufacturing in the public eye. Usually when you look at the media and when you read in the media, there always been how wonderful it is to have a new cure for cancer or whatever. And this idea of the manufacturing part, which is a very important part of the overall supply chain, it has not been very interesting to the public, but now they're aware of it. They're aware of how important it is to have the capacity to do so, but also the training of the people in place. And if you look at, for instance, UCL, since I work at UCL in biochemical engineering, it has been one of the pioneers of effectively manufacturing and providing the academic support needed to enable early technology to be translated into real products. And I think that the new initiatives which are being put in place in the UK is testimony to the fact that this is something that we need in this country. I think before all of this COVID and so forth, I think we had only 2 places. I'm not talking about the contract manufacturing setups where they have to change depending on who their client is, but more in terms of the vaccine base. We had 2 places, and I think one of them was just fill and finish. And now we realize that we really need to have that capacity in-house. I don't know whether I answered your question, but I hope I've got some way to answer it anyway.
I certainly enjoyed hearing your response. I think there's a lot to learn from that, and it'd be great to hear more from you and other experts like you to hear how we can invest better for the future. to support improved UK manufacturing. Moving towards questions that focus on developing countries. If I could ask Jusui Dong, what kind of technology and related support might be needed to de-risk or increase sustainability of vaccine manufacturing in developing countries? And who should be investing in this?
Very good question. Manufacturers in low and middle income countries face challenges in the local production of a vaccine. So many of them lack capacity in meeting GMP requirements and in receiving and absorbing technologies for vaccine production. There is also a need to ensure that a cost competitive vaccines is produced. This will help to ensure long-term business viability and retention of a production capacity. So technologies and support relating to these areas in particular would be of significant value. For instance, automating production processes reduce the risk of human intervention and error, therefore could help companies to meet the quality standards. Technologies like support flexibility of a production, for example, inclusion of vaccines and biologics manufacturing, this feeds into the utilization of a capacity, therefore ensure sustainability. And the vaccine manufacturers in low and middle income countries could, of course, benefit from these mRNA technologies, as it has advantage over other platforms. For instance, it is rapid, inexpensive, and scalable manufacturing with high production output, and also with high efficacies, and is safe. So all this in terms of the support and investment need to be de-risked and increase sustainability. It should be suited to the local and the regional context. There's no one-size-fits-all approach. Already, regarding who should invest, I would like to say all relevant actors have a role to support and invest in this development. Low and middle income countries can be very much impacted economically and socially by pandemics, and with a long recovery timelines. So government could invest first. This can be considered as insurance against future pandemics. Money invested now could provide return in the future. Manufacturer, of course, can invest related like GMP facility or this during this process and activities. Universities and research program could provide strong support on the need-based innovation, could train personnel. In international organizations like WHO and other organizations can invest and play a role based on their experiences, expertise, and mandate. Of course, donors, very important, could also invest. As the COVID-19 tells us, no one is safe until everyone is safe. So it is fundamental that support and investment are provided in a holistic, strategic, and collaborative manner. So with that, I over to you, Ben. Thank you.
Thank you. What a comprehensive response. Thank you so much. In the interest of time, we need to be drawing the Q&A to a close, but I want to respond to a number of questions that have popped up in the Q&A, particularly on technology transfer to African countries. And I'll read this out as Professor Sarah Gilbert has responded to the question, what would be the main challenges for technology transfer to African countries? And Sarah has mentioned that the new technologies require different manufacturing infrastructure to older vaccines, but new equipment can certainly be installed if the funding is there. There must also be investment in ensuring that the quality management system is up to date and lab capabilities for all in-process and release testing is there as well. So thank you, Sarah, for helping to respond to that question. Could we hear very briefly, 20 seconds each perhaps, from Dr. Dat, Manuel, and Neni, as to what have you received from working within the hub system that you would not have otherwise? Dr. Dat?
Okay, thank you, Ben. And I think that from the hub, we can get that kind of, firstly, that kind of new technology, new platform. It's so important to us. We should apply for easy to develop the kind of new vaccine, and at least for that kind of pandemic vaccine and COVID vaccine right now. For that kind of platform, we could not work before, but right now the people here can master that kind of technology by the vector vaccine. It's so important to us. And at least we can apply that kind of vaccine to develop the kind of new vaccine. Like Mrs. Dong had talked about that kind of sustainable, for that kind of sustainability for the vaccine manufacturer is so important. But I think that technology is so important also for the vaccine manufacturer in the developing country.
Thank you, Dr. Dat. Neni, may I ask you the same question? Please, just 20 seconds perhaps.
Okay, thank you, Ben. So the collaboration with VaxHub is very essential. That open the access of the vaccine technology and global network. And also for biopharma, the knowledge transfer from the VaxHub help our scientists to improve their fundamental knowledge for process operations and grow our confidence in troubleshooting and exploring new methods for the optimization beyond standard our practice. So this is building essential expertise within our company. And furthermore, I also get the insight in efficient optimization methodology that we got from the VaxHub using downscaling approach that can reduce significant costs for the vaccine development. Of course, this is very critical consideration of our region, considering the region that we supply. I think that's from me, Ben.
Thank you, Neni. Mano, last word, please. Very, very brief if you can.
Basically, I would say here actually what we learned basically yeast expression system. Basically, with our partnership with the Karen Lab, we are successfully acquired the yeast expression system to produce BLP and other products. For the initial training, actually we developed a dedicated established team at Incepta, which are now capable of work with the yeast expression system. So basically, here actually we really acknowledge the candidate support from the Imperial Hub. So without their support, we couldn't be able to so easily to establish such platform at Incepta. Thank you.
Thank you, Manuel. And thank you to all the speakers and panelists that have joined today's session. Very interesting, and thank you so much for your contributions and collaborative spirits. Happy to move on to session 3. How can we better prepare for the next pandemic? Future innovations in vaccine manufacturing. But first, can we show the results of the last poll? So with 50% of the responses have been voted for know-how and skills as the biggest barrier to vaccine manufacturing in developing countries, followed by IP barriers at 19%, access to raw materials at 14%, and regulatory challenges at 10%. Thanks to everyone for voting. It's really interesting results there. So I'd like to hand over to our first speaker of session 3, Matthew Ducharts. He's Chief Executive Officer of VMIC, that is the Vaccines Manufacturing Innovation Center. Matthew's in an executive office with over 30 years of experience in biotechnology. He has worked on a wide variety of vaccine and therapeutic platforms, as well as medical countermeasures for multiple indications, including anthrax, plague, Ebola, smallpox, Marburg, dengue, Zika, as well as radiation and chemical agents. So off to you, Matthew.
Thank you. Thanks for that introduction and for the invitation to speak today. I apologise slightly in advance if there's a little bit of background noise. The trouble with working from home, of course, is you can't predict when the council is going to start cutting the grass verges outside the window. So the tractor's gone past twice so far. Hopefully it will not go past again, but we'll see. Let's just check if I can get the slides to work. Oops, no, oh, yeah, there we go. So a little bit about VMIC to start with. So VMIC, the Vaccine Manufacturing Innovation Centre, we've been set up as a not-for-profit company, and we're there primarily to help vaccine developers really through collaborations. The facility itself, the capital to build the facility, is being funded through the UK government, particularly through Innovate UK. And the facility is really, it forms part of the national scientific infrastructure that's been put in place through the UK government to support life sciences and in particular vaccines. However, contrary to popular belief, VMIC was not established as a result of COVID-19. It actually predates COVID. So back in 2018, we were funded by UK Innovate to build the Vaccine Manufacturing Innovation Centre. And this was set up really through three founding organisations, three universities: Oxford University, Imperial College, and the London School of Hygiene and Tropical Medicine. And we were supported by three really leading players in the vaccine industry. So Cytiva, Johnson & Johnson through Janssen, and Merck MSD. So we have a good stable of vaccine experience, and our time sort of predates, as I said, COVID-19. However, we did get a considerable amount of acceleration funding during the course of the last year. So as the COVID-19 crisis sort of started to unfold, and it became fairly apparent that we were lacking some of the capacity and the idea of VMIC, unfortunately, that it was going to be a year or two too late for the COVID crisis. So the government came along and really said, well, first of all, can you make it a bit bigger so it can actually cover more than its original intent of just a few 2 to 3 million doses in the case of an emergency, but to be able to cover pandemic. So we increased the scale and capacity by about 20-fold. And also, can you accelerate that program and bring it forward? So that's what we did during the course of the last year, and we're now sort of well on with the build, and we'll come on to talk about that in a second, and when the facility will start to come online. In terms of, so what is the purpose of VMIC? What are we here to do? As I think I mentioned right at the start, we're there primarily to help vaccine developers with that sort of transition, particularly in the early stages, out of discovery and on towards the clinic, and then that translational science as you go through and on towards commercial scale. So that whole, how do you turn what's a good scientific discovery into something that can be scaled up and would be acceptable for use in humans and can be industrialized and commercialized. So that was always originally our sort of primary purpose. But we're also there to provide a level of resource and knowledge and expertise. So that is to help drive innovation. So we're there to look at ways to improve efficiencies, find new technologies to be able to develop the way that processes are made, products are made through processes. We're there to provide guidance out to not just to industry, but to anyone who's developing vaccines, whether it's academia, whether it's startup companies, whether it's large pharma. We're there as a resource to help provide guidance for that. And thirdly, we're there to provide a level of training as well. So we're there to help essentially broaden and deepen the skill sets within the workforce in the UK, in the life science sector. And that training can extend out to bringing people into the facility, as well as providing training out to people outside of the facility. And then the third purpose, as I mentioned earlier on, is really to provide a level of emergency response and now emergency pandemic response. So as I mentioned, the capacity of the facility was significantly increased from being able to make about 3 million doses of vaccine in a 4 or 5 month period through to being able to make 70 million doses in the same period. So a significant increase in our capacity there. In terms of the facility, so you can see here a picture from actually earlier this year. So the outside of the facility is complete. We're now into the fit out of the facility inside, putting the clean rooms in place, all the sort of heavy fixed equipment, and then moving on towards qualification later on this year. And the facility itself is fairly sizable. It's a 74,000 square foot footprint, so which means absolutely nothing to me until somebody said that's the size of a football pitch or a rugby pitch, depending on what your sporting preference is. So that gives you sort of an idea of the size of the facility, and it goes up for 3.5, 4 stories, so it's quite sizable. And in terms of its capability and what we'll be able to do, so it's really an end-to-end capability, everything from very early stage cell banking, viral banking, development of and manufacture of early preclinical and clinical grade material where we're making perhaps a few hundred doses, right through to scale up to commercial scales of being able to make literally sort of tens of millions of doses. So we do span the whole range from very early, very small, right through to large industrial scale. And the other thing about the facility is it's been designed to be technology agnostic, so that it's not just focused on one particular modality, but we'll be able to do RNA vaccines, DNA vaccines, viral vector vaccines, recombinant subunit vaccines, conjugate vaccines, you name it. We've deliberately designed the facility so that we can work on all of those types of technologies and all independently as well. So we are able to work on several different types of vaccines and vaccine platforms simultaneously. Okay, and I think the other thing to mention here is that so the drug substance, we have a series of suites for manufacture of the bulk drug substance itself that range from small laboratory, almost bench scale for that first in man type of studies, right through to very large sort of industrial scale drug substance suites, and similarly on the fill finish capability. So it's not just about making the bulk vaccine, it's also about filling it. We're able to fill anything from just a few small number of doses for early studies, right through to large scale industrialized production of millions of doses. Okay, so a little bit about the timeline of where we are. So right now, as I mentioned, the facility is being built. So the building is in place, the clean rooms are being put in place, the fixed equipment is starting to go in. We're really looking for the facility to complete all that installation, or the majority of it, through this year, so that we'll be able to open this year with a view to coming online and becoming operational through the course of 2022, and we'll do that in a phased way. And then once we've really established the capability and the capacity there, is then about consolidating on that and making sure that the manufacturer is able to run, and then moving on from there towards being able to do innovation and improve processes as we go forward. Just briefly, a little bit on COVID-19. So we were also quite closely involved with the COVID-19 effort, to not only just in terms of expanding the facility, but also helping to set up a rapid deployment capability. So we actually realized that our facility wasn't going to be ready in time to manufacture vaccine when it was needed for the country over the course of the last year. So we helped to buy our equipment early and worked in partnership with another organisation, Oxford Biomedica, in fact, to help set up a capability there using our equipment in their facility, in their clean rooms, to be able to provide the country with vaccine earlier than anticipated, and that was approved back in October of 2020, which meant that manufacture of vaccine could be completed, ready to go out into people at the end of 2020 and early 2021. And then conscious of time here a little bit, my last slide, I did want to say a little bit about working with universities and specifically what we've been involved with there. So as I said, we're setting up now, so we're not sort of we haven't got everything up and running just yet, but what VMIC will do is it will provide capacity in particular to help with that early transition out of discovery, where a lot of the work that the academics and universities are doing great stuff with new vaccines that are coming through, and we've heard about some of those today. So what we'll be able to do is help to do that transition and get that those products out and made to GMP and into the clinics early, and in an expeditious and an economic way. We'll be able to provide a level of expertise out to help developers and academics, particularly sort of identify early on what are the right sort of processes that are industrializable or scalable, what are the types of cell cultures or vectors, media components, separation technologies, whatever it might be. Some of those things that you do in the lab are not necessarily easily scalable, so it's important to identify those early on, and we can help provide some of that and to sort of navigate that sort of myriad of different potential pitfalls of going down the wrong path. And lastly, I think that VMIC is able to help kind of close the loop in the sense that academia produces a lot of not just products and from product development perspective, but also new ideas and innovation that comes through and is taken over by industry. And what VMIC can do is help sort of close the loop back to academia by taking those industrialized products, processes, and then training and providing knowledge and information on how that's been used within industry back into academia, so they're aware of that. And I think with that, I will draw to a close, as I may have overrun by a few minutes, so I apologize for that. Thank you for your attention.
Well, thanks to you, Matthew, for such an interesting presentation on VMIC, which of course we're all interested in and supporting in some way. Before introducing our next speaker, apologize to Parissa Masami. She's our early career researcher assisting in selecting questions for the panelists at the end of this session. So please do enter questions in the Q&A session at the bottom of your Zoom screen. It's now my pleasure to introduce Mike Whelan, currently project leader for CEPI, the Coalition for Epidemic Preparedness Innovations. Mike has a very large portfolio within CEPI that includes vaccines for Rift Valley fever, platform technologies, and certainly more recently for COVID-19 projects. So Mike, very interested in hearing your talk. Off to you.
That's brilliant. Thank you very much for the invite. Okay, here we are. Yeah, for those of you that don't know now, hopefully by now you do know who CEPI is. But when we started, nobody really did. Sarah hit the nail on the head this morning, really. There was a lot of fallout from the Ebola outbreak, and actually CEPI was one of them. And the whole idea was this should never happen again. And of course it did, at a much bigger scale. But the idea was to set up this global partnership with the lofty goal of making epidemics and pandemics no longer a threat to humanity. Now, clearly we haven't got there yet. But having a coalition like CEPI is really quite unusual because it means we interact with everyone. We're non-profit making, but we work with pharma, we work with academia, regulators, governments. We work with everyone. And the C in CEPI stands for coalition, and that's a really important thing for us. Now, when we started, we were working very much along the lines of the WHO blueprint list for diseases which could cause an epidemic, and frankly, nobody was doing anything about it. So we started with Lassa, Nipah, and MERS. And it was a good starting point. And, you know, we were fortunate enough to work with both Robin and Sarah on these, and it was great because we managed to get ahead of the game a little bit. We also did some platform technologies, and I absolutely take on board Sarah's comment earlier on. Platforms are key to this. We would like to have had a little bit more time to develop those platforms, but unfortunately COVID-19 didn't read our project plan. So we tried to get ready for it, but we could only do what we could do. We extended, after the first three, we extended into chikungunya and Rift Valley fever. Rift Valley fever is one of my personal favorites because it's a true zoonotic disease. And let's face it, pretty much all of these are zoonoses. So unless we can start to identify these things at source, there's not much we can do. Now, the comment was made earlier on about MERS. We would have liked to have done that, but at the time we didn't have the budget, but it's certainly something we're thinking about in the future going ahead. But anyway, with all of these in the background, COVID-19 hit. And we worked on 12 candidates, of which 10 are now in active development. Now, some of that early work that we did on MERS in particular, and Disease X with Sarah and Robin and so forth, was actually really helpful because we co-funded it. Obviously, the work was going on anyway, but it was really helpful to know that we could get a march on things. Things like dose selection was actually done before the main phase 1, phase 2s were done, and that really moved things forward. We established probably one of the largest vaccine portfolios in the world for COVID-19, but we've done a lot more than that. Things like harmonizing, how do we know if they work? Tracking new variants. We're working on broadly protective coronavirus vaccines for a next generation, if you like. But through all of this, the most important thing to us, of course, is equitable access through COVAX, and I'll come back to that. Just to show you of what we've supported, we started off with DNAs at Inovio. We sent some seed funding to Moderna and CureVac on RNAs. Viral vector, well, obviously the lead one was Sarah's Oxford team. We funded with AstraZeneca, we funded the procurement of 170 million doses into COVAX. And I've been working with them for the last year, and they've just been incredible. We mentioned earlier on about intranasal. I'm working with a project in Hong Kong where we're looking at a nasal installation of, it's actually an influenza virus carrying the RBD. Very interesting. It's going to be very difficult to license, so we'll just have to see how that pans out. We've also been involved with things like Clover and Novavax, which I'm sure you've heard about, but we've also done some work with some new companies like Biological E and SK Bio and VBI, which is a virus-like particle. And these last three are actually new variants. So we have already started developing vaccines based on new variants. Most people seem to be using the South African strain. But I also wanted to touch on some of the things we've funded in enabling science, because it's all very well everybody doing things independently, but we do need to know, are they comparable? So things like we funded an international reference standard for SARS-2. So everybody's using the same serum with our friends at NIBSC. We've been looking at epidemiology. We've been looking at sequencing. We've set up the centralized laboratory where all of the labs in the network will use exactly the same technique. They will use the same SOPs, they will use the same reference standards, they'll use the same methodologies. And again, this is just to make our developers able to compare the results and say, is this working or isn't it? We've also been working with GISAID and NIBSC to track new variants, and we've got a funding call out at the moment for about $140 million to generate clinical data looking at new variants. So we're constantly watching what's going on. Now, coming back to COVAX, COVAX is probably one of the best things that we ever did. It's the only solution which will allow fair and equitable access to vaccines, regardless of the ability to pay. We're aiming to have 2 billion doses into COVAX at the moment, or by the end of 21. And what we heard from President Biden this morning is very, very great news. But we've also worked with a number of developers. Probably most of our doses have come from AstraZeneca so far. But it just shows that a non-profit making function will work. We have got doses out so far to at least 129 economies. This is rising by the day. We have over 80 million doses delivered into COVAX, all at non-profit. I mean, this is just incredible. So, looking at the future, COVID-19 has really changed the landscape. There now, as I think we've all realized, there's the political will to invest in health security. We just heard from Matthew about things like VMIC. I'm not sure they would ever have got the funding if the political will hadn't been there, but now it's there. There have been revolutions in technology, things like the new mRNA vaccines, incredible. These things didn't even exist 5 years ago, and it's just amazing. And there's also the global desire to say this can never happen again. But we think developing a global end-to-end R&D system and financing model is the only way to do it. So COVAX, although it's targeted at COVID-19, we believe it should stay. And this is something that we would like to use as a model going forward. Now, one of the things they asked us to think about was reflections on academic and commercial partnerships. And it's a very big idea. So obviously in the interest of time, there's only so far we can go with this. But just some rough ideas, like where do the ideas come from? Well, it's a combination. You know, academia obviously is a source of ideas, but a lot of the big companies were working on platforms as well, and they were able to turn those over. Now, things like the maturity of platforms, developing them for other projects. I know this is something Sarah said earlier on, and I completely agree with her. So really developing everything there is to know. Things like there is no such thing as a platform master file in a regulatory setting. We would like to change that. Why not? Things like clinical trial methodologies we've seen in the current COVID-19 outbreak. People tend to have been combining phase 1 and phase 2, and that makes a lot of sense. They're doing things like dose optimization in phase 1s, which they never did before. And it's really speeded things up and moved things forward. It's not at the risk of safety or anything like that, but they've just made the system better. Technology transfer, this is a big one. And I've put here the lines are blurring. And what I mean by that, and I think what you guys have been doing here in the vaccine hubs is actually case in point. If 5 years ago we'd gone to most academics, we'd have had a bit of a blank expression whenever we mentioned the words GMP. Now we're getting things out of academic labs delivered properly. I mean, case in point, when Sarah transferred over to AstraZeneca, and I know because I've seen it, the package they got was, that's a commercial package. That's how you do it. And it saved so much time. And this sort of thing we really have to embed now going forward, because it is the key. I mean, I've always been of the opinion that in the lab, if you're not using an industrial process, you're frankly wasting your time. So this was great to see. That said, there are certainly problems remaining. The geographical footprint of where the manufacturing facilities are. We've heard from some of our colleagues in the low to middle income countries, there's not enough of them. We need to have these facilities ready. We need to have them available all over the world. Production in NELMIX, we need to be funding that. We also need to have facilities ready. Where do we get the supplies from? We know there's a worldwide shortage of supplies, and in fact, CEPI is currently trying to do a marketplace where we can share supplies between manufacturers. But this sort of thing is going to be a major problem continuing. So, I mean, obviously, it would be fantastic if we could have a hot facility, a facility that was always on. VMIC may be just such a thing, but one is not enough. We're going to need more. And who pays for it? Who keeps these things hot? Who keeps them going? And of course, equitable access is central to everything we do. We want vaccines for everyone, regardless of the ability to pay. Now, I'm sure you've all seen this graph, but I think it's an amazing picture. In COVID-19, it took only 300 days from virus characterization to submission of phase 3. That's amazing. CEPI comes to the end of its funding cycle at the end of 2021. So we're going to launch CEPI 2.0 next year. We want to change that to 100 days. How do you do that? Well, we think there are 3 pillars. Prepare. We've got to be ready this time. We've got to develop vaccines for known threats. They've got to be ready. They've got to be stockpiled. They've got to be developed. We've got to transform. We want to make a library of vaccines for families of viruses so that we're ready when something similar comes along. Can we compress that timeline down to 100 days? It's aspirational. If we don't make it, it doesn't really matter, but at least it'll be faster than anybody's ever done it before. And we want to connect. We want a global network of lab capacity, manufacturing capacity, preclinical, clinical, all of that needs to be ready to go because this will happen again. And I think from what we've heard today, UK academia and industry has shown, you know how to do this. Let's do it together. Thank you.
And thanks to you very, very much, Mike, for such an excellent presentation on your work and all of the activities from CEPI. May I ask for poll 3 to be shown on the screen? So just please do take the time as we're handing over to new panelists to please answer this question at any time during the Q&A session when time does allow. But I do want to thank the speakers, Matthew and Mike, for their excellent presentations and introduce 2 experts as part of our session 3 panel. First, Professor Gordon Dugan, Head of Pathogen Research and a member of the Board of Management at the Wellcome Trust Sanger Institute in Cambridge. He's also an honorary professor at the University of Cambridge and a fellow of Wilson College, Cambridge, also a massive contributor to Wellcome Trust and their goals, and a recent winner of the Albert B. Sabin Gold Medal. So congrats, Professor Dugan. Also like to welcome Professor Brendan Wren, Professor of Microbial Pathogenesis and Dean of Faculty of Infectious and Tropical Diseases at the London School of Health and Tropical Medicine. So Parissa is helping to sort through questions that are popping up. And I would like to begin with a question for Gordon to ask what lessons can we learn from how the UK has restructured how the UK has restructured the manufacturing sector in the 20th century. What can we learn from that to inform us on how to move forward in a better way in the 21st century?
Thanks, Ben. So I think I'm probably one of the few people alive who was working in the vaccine industry when we were completely autonomous in the UK. We made all of our own vaccines. We had discovery, development, clinical trials, regulatory manufacturing all on site, and we exported. And that was through the Wellcome Foundation and GSK. And we went through this fallow period from around 1990 to more recently, where the only people that stayed in the game were the people, you know, the Jenner in Oxford and some people at Imperial. And I think one of the standouts to the Jenner and to Adrian and Sarah is not because of what they've done this year, but it's more about what they did for the last 20 years. When they had the resilience and guts to stay in the system, which was not working for them. I think that was one of the points I'd quite like to make. But when you think about, you know, what we should do next, so I've got a little tick box. I've got a tick box against discovery. I think we're very good at discovery in the UK, very innovative, particularly with my work at the Wellcome, I've seen that. Development, you know, we've got VMIC and we've got Oxford, and we've heard from Mike about CEPI. I think we're very, you know, capable in that area. We could probably expand it down at Portland places. Clinical trials, without doubt, were excellent. You know, the UK has got a tremendous reputation now, rightly, for the work they've done on trials. And regulatory, I've been working at the MHRA for the last several months on their expert panel, and I can tell you the MHRA is fit for purpose and ready to, you know, it's a high quality regulatory organization. It's the question then is about manufacturing. It's quite an interesting one, really, because ironically, we've had one of the biggest vaccine manufacturers in the world, GSK, in the country for the last 50 years, but they manufacture abroad. They manufacture mainly in Belgium, and it's probably a big part of the EU legacy. And they've got an enormous city almost making So we wouldn't want to replicate that, but what we could do, our companies like GSK, but also interestingly AstraZeneca, are they going to start to manufacture more locally? And there's lots of other companies coming through that we're looking at in the MHRA who are bidding to introduce vaccines, Moderna, for example, into the UK, and will they start to manufacture? And the other thing, will the government participate? So we could start to manufacture our own vaccines through a private or a public enterprise. We have a choice. And the last thing is, which vaccines will we manufacture? Will we manufacture every vaccine, children's vaccines, flu vaccines? We do manufacture flu locally, for example, in Liverpool. So the question then is, what vaccines would the UK want to manufacture? And the last point I'll make is, I think for people listening in from LMICs, you will hear many, many reasons why you cannot manufacture locally. We've heard some of them, the IP barriers, the lack of expertise and lack of skills. But what I would say to you that you must battle through all of those barriers if you're ever going to actually be autonomous and be able to manufacture locally, which I think is needed now after we've seen the problems of vaccine inequity. Do not be persuaded by people who say we cannot do this because of X, Y, and Z, because if you think about all of the barriers, you'll never get through them. And I would encourage and the Wellcome Trust and other organizations that we should facilitate and try to help local manufacturing capacity be built, but also the capacity of investigators in discovery, development, trials, regulatory, and all levels of vaccine manufacturing to be built across the globe. That's my speech. But I'm happy to now hand over, because of time, to other people. And also, as you know, Ben, I've written a blog on this. So I have a blog. If you do go gdugan.blog, you'll find a little blog on this area, which you might find time to read if you're interested. Okay, thank you very much.
Well, thanks for your response, Gordon. I also encourage people to read anything from you, because it's always informative and it's just excellent resource.
And can I just make one final point? I want to thank the hub itself, you, Ben, particularly, and all the hub people for allowing me to participate. It was preemptive, really, and in preparation for what's happened in the last year. So thanks for that.
Well, obviously, it's returned. Massive thanks to you and all of the other vaccine experts and researchers involved. Thanks for everything. You mentioned clinical expertise and success in the UK, and that brings me to my next question for Mike, which is, are we at the precipice of being able to change vaccine clinical development? Can we create more standardized ways of merging phase 1, 2, 3? Are we stuck to how that's worked over the past few decades? What are our options in moving forward there?
Well, it's obviously going to be a stepwise process, and obviously safety is paramount. But I think the regulator, and certainly Sarah alluded to this earlier on, the regulators have been very open to having rolling submissions and saying things don't have to be the standard 1, 2, 3 paradigm that we've had previously. Also, the other thing that we noticed is a lot of the preclinical toxicology studies for COVID-19 were actually done in parallel with the phase 1. That's a real sea change. So I think it'll come with time, but we can't do everything at once. And if we were, that would be far too risky. But I think the conversations are there, and I'd be interested to hear if any of my colleagues think otherwise. But certainly I think the regulators have been very open, but they look at safety number 1, and we totally agree with them.
Thank you, Mike. And we should all thank the regulators, based in the UK, particularly the MHRA, as Gordon has pointed out as well, have been massively more than special in carrying out all of the important activities over the past year. So thanks to everyone from the MHRA in particular. A question for Matthew. Now this comes from the Q&A section, but I'll expand on it a bit because it was an early question in the program, which is how can the UK manufacturing capability be built? So in particular, how could the VMIC contribute in a better way for global health security through its infrastructure investments from the UK government?
Thank you. And it's a great question. So, I think probably the first thing to say is actually let's have a look at what we've actually done so far. So the UK really has demonstrated its ability to be able to rapidly respond to the pandemic. Was it perfect? No. Did highlight some gaps in for example, in capacity and infrastructure. But nonetheless, let's not sort of forget what actually was achieved. Went from having things we heard earlier today, a sequence became available in January, and by October we were mass producing vaccine tens of millions of doses. So we had the ability to do it, but I think there are lessons to be learned and things that we can do and improve on. And one of the things, VMIC unfortunately was not built at that time, so we weren't quite ready. But one of the things we will have going forward is as VMIC comes online, is to actually have some of that capacity and capability, not just to be able to manufacture for the country on a tens, hundreds of millions of doses scale, but perhaps even more importantly, it's got the capacity to really facilitate that early transition out of discovery and into the clinic. One of the things that I heard on innumerable occasions from people was how difficult it was for people to be able to access facilities that can make GMP material, but where they only needed a few tens of doses or 100 or 200 doses. Everyone was, all the sort of big manufacturers were like, oh yeah, if you want to make 20 million doses, we can do that. If you want to make 200 doses, not really interested, because it's just not worth it. So one of the things that VMIC will help to do is to sort of bridge that gap and provide a facility whereby people can, early developers, academics, anyone with sort of early stage programs will be able to, quickly and rapidly manufacture material to be able to go forward onto the clinic. And I'd also reiterate a point that Mike made a little earlier on about the importance of using industrial technology to be able to do that. So don't just go and do something that's expedient and easy to do it in the lab if it then means that we then can't industrialize it. So use the knowledge that's out there now to use platforms, media components, separation technologies, whatever it is, make sure you're using things that are scalable, that are industrializable. And that again is something where VMIC will very specifically be able to help.
A new question has popped up with the help of Parissa, which is asking whether there's a model whether VMIC is a model that's being used for other countries. For example, is it a model that can be used to establish similar infrastructures in African regions?
So I think the short answer is yes, and actually there has been considerable interest from a number of different countries, from in particular Africa, Latin America, Asia, countries have all approached VMIC and the UK government about trying to find out more about how VMIC was set up, how it runs, and could they do something similar in their regions. And I think there's absolutely an opportunity to be able to do that, and I think that's something that we can help do as well. We're not here to be overly competitive or anything. We're a not-for-profit company. We're here to sort of help the sector move forward. So that's absolutely part of what we're here to do.
Thank you, Matthew. Turning to Professor Brendan Wren, what lessons can be learned from global manufacturing and distribution of COVID-19 vaccines during the pandemic? How can we be better prepared for the next one?
I think there are two major lessons from the COVID pandemic. Firstly, we do need vaccines for all, and that we need more than one or two vaccine platform technologies for better coverage. And in terms of vaccines for all, I think COVAX is a shining example, but we do need local manufacture, and it's very heartening today to see the presentations from Vietnam, Bangladesh and Indonesia, and certainly India leads the way in this. I noticed there's some questions about what about sub-Saharan Africa, and I'd like to highlight the work done at the Institut Pasteur in Senegal that I've visited a couple of times, and their vaccine production program, Yellow Fever, is really excellent, and they're expanding this through a new science park outside Dakar. So I think there's real encouragement there. The biggest driver for vaccines for all is inevitably lower cost, but I think there's real encouragement here from several new technology platforms being developed to reduce cost and to make these processes simpler. So from my own experience, this includes making glycoconjugate vaccines in E. coli, which we've been developing for 20 years now, whereby the E. coli cells, they act like mini factories for their single-step purification of glycoconjugate vaccines. So it's a simple plug-and-play system that can be transferred to low and middle-income countries. And we are really indebted to VaxHub for getting us to the next stage in terms of upscaling this technology for these platforms. In particular, this has been against pneumonia, but for other vaccines as well, including multi-drug resistant bacterial infections, are also in the pipeline. We do need to have more than one vaccine platform technology. We've heard a lot about the RNA and adenovirus approaches that have been very effective, but we do need to continue to develop more novel platform technologies. Fighting pandemics, it will be a case of horses for courses. And I am quite often asked about the next pandemic, but I would argue that we are actually in the midst of a slow pandemic. Antimicrobial resistant bacteria, for which we have no vaccines, is a major, major problem. And it's a huge current threat and future threat as well. So we do need to develop vaccines, beyond vaccines, beyond viruses that cause acute pandemics. So I think overall, we're really in exciting times. We're going through a real golden era for vaccine development and production, with several new technology platforms being developed that will mean vaccines for all. And there's never been a better time to be in vaccinology. And now we have significant public and political support. It's up to us to take the opportunity with the appropriate support.
Well, thank you, Professor Wren, for that. I'd like to thank you and the other speakers, panelists of today's session 3 session. Parissa, massive thank you for your support today as well. And I would like to thank Sarah and Robin for answering so many questions everywhere on the Q&A and chat section. It was very, really helpful. Can we show now the results from the poll 3 question? So the strongest, the highest number of the responses goes to stronger collaboration and network creation, for which of the following should be given highest priority to ensure better pandemic preparedness at 55%. This was followed by local vaccine production at 30%, training and upskilling 10%, better infrastructure for vaccine distribution at 4%, and at the bottom, formulation and stability studies. So thanks to everyone for participating in that poll. It's then now my pleasure to hand over to my colleague from the VaxHub, Martina Micheletti, to chair session 4.
Yeah, thank you, Ben. So just in the interest of time, we have asked Robin and Sarah to make some final comments and wrap up some of the points really made throughout the meeting. So Sarah, if you'd like to start.
Thank you. Well, I'm not going to take very much time. I think what we've been trying to present to you today is the way that we've worked together to create networks, to bring together different expertise, to bring together different types of organisation, from academia, where there's often the innovation, to industry, the implementation, with a lot of small companies involved in our hubs as well. We really want to keep these activities going as we reach the end of our current funding. We feel it's been extremely valuable in responding to the current pandemic, and we could do more to improve our resilience for the future. And I'm sure Robin has some other thoughts to add on that.
Thank you, Sarah. So I'll be brief as well. And I think it's important to recognise that the funding of the hubs was very well timed and very innovative. It came out of ODA funding, and so it was limited to 3 years, and that was what was available at the time. But as we look at other manufacturing sectors in the UK, it's very clear that the other approaches in areas of strategic importance have set up manufacturing hubs that have run out for 7 years. And it's that kind of long term of investment that I think is essential to develop the new technologies and really skill the UK and the UK partners for the future. It's clear that we have excellence in research and discovery. We have all the different components in the UK, whether it's academic groups, whether it's agencies like Catapults, VMIC, CPI, many others, but having a way of coordinating and keeping them all engaged would be important. And I think the danger is that there's always ability of building things that don't get used, whether that's building manufacturing facilities in parts of the world that aren't sustainable, or whether that's building manufacturing expertise in the UK. And so Mike talked about keeping things hot lit. And one of the things that I think we recognize in some parts of the world, a classic example would be the DARPA initiative in the US, is that you often need something to drive things through and keep them active if you want to absolutely synergize and exploit maximal benefit from the infrastructure that's there.
Thank you, Robin. I definitely second what you have said regarding the length of the hub and the need for longer term investments, also to support some of our interesting SMEs work, but also to really nurture longer term collaborations with some of our developing countries partners. I'd like maybe to finish with some remarks. So thank you to all the speakers for presenting their work, panelists for their contribution. We are aware some of the attendees, I think, had challenges with the registration link. We investigated this a little bit in the background. It seems this was a Zoom issue, but for this and other reasons, I really hope that our next event is going to be face-to-face, really to maximize the network and possibly have another opportunity to bring the community together. Behind the organization of this event and behind the scenes today, there is an amazing team managing a number of different aspects. So I'm very lucky to work with them every day. And we're now planning some post-event dissemination with Ben. So we will be in touch.